This is for their information that there is no such WhatsApp group. There is no such WhatsApp group.
This is for the information of the postgraduate students that there is no WhatsApp group being created to share the files or the PPTs of all the speakers through these 3 days. There will be an alternative that will be arranged. The discussion is still going on with the technical team and the organizers. How we can make these files available to the students. Please do not post messages saying add our number to the WhatsApp group. I repeat, there is no such concept of creating a WhatsApp group. Thank you. Dr. Lakshmi, have you joined?
Good morning, ma'am. I have joined.
Oh, good morning, Dr. Lakshmi. Dr. Lavanya is there. I can see her.
Good morning, Dr. Lavanya.
Good morning, madam.
Another 3 minutes. I shall start. Okay, we have to wait for 9.
Yeah. Hello. Good morning, ma. Yeah. ಏನು ತೊಂದರೆ ಇಲ್ಲ. ನೋ ಆ್ಯಕ್ಚುಲಿ ಏನಾಗಿತ್ತಂದರೆ ಇದು ಪೆರಿಯೋ ಪನೋರಮಾ ನಡೀತಾ ಇದೆಯಲ್ಲ, ಅದಕ್ಕೆ 2 ಸೆಷನ್ಸ್ಗೆ ಮಾಡ್ರೇಷನ್ಗೆ ನನ್ನ ಚೂಸ್ ಮಾಡಿದರು. ಸೊ ದಟ್ ವಾಸ್ ಟುಡೇ. ನೆನ್ನೆ ಐ ಗಾಟ್ ಬಿಸಿ ಟು ಇನ್ಫಾರ್ಮ್ ಯು. ಸರ್ ಅದಕ್ಕೆ ಆಯ್ತಾ
Yes, sir. I wish a very good morning to the senior faculty members, speakers, moderators, teachers, and the students. We begin today's program We have a set of 6 lectures for today. The first lecture is from Dr. Lakshmi. The moderator for this first session is Dr. Lavanya from Ramaya Dental College. I now call upon Dr. Lavanya to take over this session. Thank you, Dr. Lavanya. Dr. Lavanya?
Hello. Yeah. A warm welcome to one and all, to our senior professors, teachers, and our colleagues, and my dear students. I welcome you all to this first session of the Perio Panorama, and it's a proud moment that I introduce Dr. Lakshmi to all of you. She has completed her Bachelor of Dental Surgery from Government Dental College from Kerala, and she has completed her master degrees in periodontology from DAP RV Dental College, and she is presently pursuing her doctoral program, and she has her research interests are in non-communicable diseases, local drug delivery systems, phytonutrients to address periodontal disorders, regenerative therapy in horizontal bone defects, novel microbial therapy in aggressive periodontitis using bacteriophages, and improvisation of periodontal diagnosis and instrumentations. And she has many publications to her credit in both national and international journals. With these few words, I welcome you, madam, and I hand over the podium to you. All the best. Thank you.
Thank you very much, ma'am.
Thank you.
Good morning, one and all. I thank BAP for this opportunity to present and have a session with the students on genetics and periodontics. So with this, I'll start my presentation. Is my screen visible?
Yes, Dr. Lakshmi.
Yes, thank you so much.
After you come on to the slideshow, please hide that this thing that has popped up.
That hide this thing, isn't it,
Yes,
ma'am? Yes, I should do that. Okay, I'm hiding this. Thank you.
And Dr. Lakshmi, please maintain the time. At 40 minutes, you will receive an alert in the form of a buzzer.
Yes, ma'am. Yes, ma'am. So again, good morning. So I am Lakshmi. I am from MCODS Manipal, and I'll be having this lecture on genetics in periodontal disease. So what is this genetics? So we'll have a background on genetics first. So it is a science of genes, heredity, and variation in the organisms. So it will provide the tools in the investigation of the function of a particular gene. So now we all know the genetic material of cells. It is the deoxyribonucleic acid, DNA, which contains all the information that is needed to build the entire human body. So now what does this DNA comprise of? It has this phosphate group. There is a deoxyribose sugar group, and then the nitrogenous bases. And the nitrogenous bases, you have purines and pyrimidines. That is, purines are adenine and guanine, which has a double bond, and pyrimidines, which are thymine and cytosine with a triple bond. So then we come to this term called codon, where you have 3 nucleotide sequence, which will form the codon, which is a code for the amino acid. And then we have gene. What is a gene? It is a basic unit of heredity in living organism. So it is a segment of the DNA which is located at specific points in the chromosome. So these genes, they will contain the instructions for making proteins. So in a gene which codes for a protein, we have exons and introns. Let me see whether I get the pointer. Yes. So we have exons and introns. So exon is a stretch of DNA which will be retained in the mRNA, which will be translated to protein, and this intron that will be sort of eliminated from the sequence. So now we have this chromosome where you have the nucleotide, the genes. Then you can see here something called the histone and the nucleosome. This histone, it is a highly alkaline protein, okay? So they will allow the packaging and ordering of DNA into the structural units, which are called nucleosomes. So they also form the major structural proteins of chromatin, okay, and they have this role in gene regulation. So then we have this chromosome, which is a DNA structure which is combined together to form the chromosome. So when you say chromosome, we have telocentric, acrocentric, submetacentric, and metacentric based on the position of the centromere. And then when we say some polymorphism, the location 3p22 and all that, what does it mean? It is just the cytogenic banding nomenclature. So if it is 3p22, it means it is chromosome 3, the P arm of the chromosome, and that the subband where you have the 2 location 2, and then a subband of that where there is a location of 1. So you name it as the banding nomenclature, it will come to 3p 22.1. So when you say there is a polymorphism in such a location, it means just this. That is in the chromosome, this particular chromosome at this location, you have some polymorphism. So now what is a genome? Genome, it's a complete set of chromosomes of an organism. So every organism has a specific genome. That is the number of chromosomal pairs. So for we all know, human genome has 23 pairs of chromosomes, which is approximately 3.2 into 10 raised to 9 base pairs. So now let's come to this thing, a rough, shall we say, shortened version of what is DNA as information, source of information for proteins. We have ATGC, which constitutes it's like the alphabets. The codon, it forms the word. The gene is the sentence. Chromosome is the chapter. And genome is the book. Now, what is this gene expression? See, this is all inside the DNA. How does it get expressed? The expression is when it forms proteins. So this is the process of gene expression. So first you have something called transcription. You can see here the promoter site. They are basically regulatory elements. Okay, so then we have the transcription start site, transcription stop site. Then you can see the introns and the exons. So what is this transcription? It is the synthesis of RNA from the DNA. So, and this is done by the enzyme RNA polymerase. Then, so initial transcription product will look like this. After that, it undergoes a process called splicing. Splicing is when the non-coding region of the gene, they are excised out. They are just removed out of the messenger RNA transcript, and the exons, introns are removed out, and the exons, they are joined together to create a mature messenger RNA. So this is a finished transcription product. It contains only exons. And then there is the translation. So this is when the ribosomes that are present in the cytoplasm or endoplasmic reticulum, they will synthesize the protein after from this transcription product. So that is called translation. So now you should know some terminologies. It can come as viva questions, like alleles. What are alleles? They are different versions of a gene. So dominant alleles, they are indicated in capital letters, and recessive alleles, they are lowercase letters. Then homozygous dominant means having 2 copies of the same dominant allele. Homozygous recessive, it means having 2 copies of the same recessive alleles. And you should remember that recessive alleles are expressed only when they are homozygous. Then we have codominant. So these, both of them are expressed, and they will be, they are both dominant. So both are expressed. Heterozygous, they have 2 different alleles. Then the dominant allele will be expressed. So you should know this term gene loci. They are specific positions of gene on a chromosome. So now what is genotype? It is a combination of alleles of a gene carried by an organism. And phenotype, it is the expression of alleles. So what is the physical manifestation of the particular alleles carried by the organism? So let's come to the genetic variation, which is related to the disease process. We have mutations, which are infrequent, potentially harmful variations, and polymorphisms, which are common, generally harmless variations. Okay. So that is, mutations are more rare. It is when I read through it, and it is less than 1% is considered as a mutation, and it is more than 1% in the population, it is considered as polymorphism. So now we have point mutations. Point mutations. See, this is just an example. Okay, these are not obviously coding things. Okay, suppose if there is a nucleotide, it's a normal thing. Okay, a substitution is where one nucleotide there is a change. Insertion, one additional nucleotide is there. Deletion, when one is gone, and inversion is when there is an exchange between the two sequence. So there can be silent mutations. You can see here, although this codon has changed, they code for the same amino acid. So there is no problem. Missense mutations, you get a different amino acid. Nonsense mutations, it becomes a stop codon. So this amino acid sequence, it is truncated, it stops there. And frameshift mutation here, just this one base is changed, but the entire frame changes. You see, from GAA it became AAG, then ACA, instead of GCA it became CAC, like that. Entire frame is altered. So you can use this mnemonic to remember the point mutations. Then we have the block mutations like duplication, inversion, deletion, insertion, translocation. So again, you can see the changes in the figure. In duplication, we have a duplicate here. Inversion, it is there like that. And you can use this particular mnemonic for that. And then coming to the polymorphisms. Polymorphisms, you can see single nucleotide polymorphism. Here there is just one change in the nucleotide. Then we have short tandem repeats. They are also called microsatellites, okay, or simple sequence repeats. They are basically accordion-like structure of DNA. So there are core repeated units. They may be 2 to 7 nucleotide in length, and they are repeated for about half a dozen or more times in the genetic sequence. Okay, then we have VNTR, variable number of tandem repeats. Tandem repeats just means that the repeats are located adjacent to each other. Variable number of tandem repeats, again, we'll have 2 or more base pairs that are repeated tandemly. And it is and the thing is, they may have a highly variable number of tandem repeats within a population. So these are the polymorphisms. Now, what is the significance of polymorphism? It is that it may be a functional or non-functional polymorphism. If it is outside of the gene, then it does not affect any protein production or function. But if it is within the gene, whether it is in the regulatory part or in the coding region, if it is in the non-coding SNP within the regulatory sequence, it can change the amount of protein produced. And if it is within the coding SNP, it can change the amino acid sequence. Now, there are several methods for genetic analysis to study how it is inherited, how it is associated, and all that. They include segregation analysis or pedigree analysis, linkage studies, twin studies, association studies. So you can use this particular mnemonic SPLAT to remember what all are the different forms of genetic analysis. So pedigree analysis, it's a pictorial representation of a family tree, and it will show the pattern of inheritance of a character. In segregation analysis, what we do this, there is also the statistical models will be there. So that will make alternate assumptions regarding what might be the underlying mode of inheritance, and probably the impact of environmental factors also will be considered. So there are different modes of inheritance that you may see using this pedigree analysis and segregation analysis: autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, Y-linked traits, and all that. So there are some advantages and disadvantages. Advantages is that we can find out whether the disease gene is an autosomal or a sex chromosome related, or whether it is dominant or recessive. However, they have a low power to resolve the heterogeneity. So that is, it is difficult to distinguish between the genetic and environmental influence, and obviously they cannot provide any specific information about a specific gene. Then comes the linkage analysis. So in linkage analysis, there is the logarithm of odds score. It compares the likelihood that two genes are truly linked or it is by chance. So there is this term, linkage disequilibrium. It indicates that some genes occur together more often than would be expected by a random distribution. So you can see here, This is a locus of a disease gene. It is located here. So DNA markers that are close enough to the disease gene, they tend to be inherited together with the disease gene. So this closer the marker, the closer the linkage, and more likely that they will be inherited together. For example, it is said that localized aggressive periodontitis. Please remember, in this presentation, we will be terming it as aggressive periodontitis only, because most of the studies are prior to the new classification. So we'll term it as aggressive periodontitis. So localized aggressive periodontitis, it has been found to be linked to dentinogenesis imperfecta. However, these can be used, they are mostly useful for major disease genes, and complex diseases, they are often difficult to identify because there are a lot of factors contributing to the disease process. Then we have the twin studies, where the influence of genetic and environmental factors can be estimated. So we have this concordance, that is the degree of similarity between twins in one or more characteristics. That is a probable viva questions. What is concordance and discordance? Discordance is a degree of dissimilarity between the twins in one or more characteristics. So here you can make the heritability estimate, that is proportion of phenotypic variations, which can be due to the genetic variation. So you can estimate that using the twin study. Then we have the association studies, such as the family linkage studies, candidate gene association studies, genome-wide association studies. So in the association studies, they can suggest the presence of a variant allele, okay, which will contribute to the disease process, but it does not confirm the link between the tested allele and the disease gene. However, in the candidate gene approach, you can choose a gene from a prior knowledge and then analyze the gene. And in the genome-wide approach, we'll take all the genes and see which particular gene is associated with a particular disease process. So here, candidate gene, it is more like a confirmatory test, whereas the genome-wide association studies, they are more of an exploratory test. Then we have the human haplotype map project, or HapMap project. They have been used to identify variations across genomes. So this project, it has been expanded to include about 11 human populations with genotypes for 1.6 million SNPs. So now this allows for identifying the linkage disequilibrium, which we discussed a little earlier. Now, this is a question that has come in our GHS paper. What is the role of genetics in periodontics? It has come as a long question. So this constitutes the answer to it. And so if you learn this answer, you can answer anything on genetics. That is, what is periodontitis? Models of periodontal disease pathogenesis, with a focus on those concepts which have spoken about the genetic influence. That is Convent model, biological system model, and 2015 model by Chappell. Then a note on epigenetics, because it is mentioned a lot, and periodontal disease pathogenesis. A note on genetic dysbiosis and infectogenomics in periodontal disease pathogenesis. Then the various polymorphisms in relation to periodontal disease, where you have to give the classification. Then requirements in proving the disease polymorphism association. Summary of different polymorphisms, that is a cytokine, receptor, and other polymorphisms. Then aggressive periodontitis, like I said, it is the 1999 classification system. Most of the studies are based on that only. Then in which you have to talk about the polymorphisms associated with aggressive periodontitis, taken from the summary of different polymorphisms given earlier. Then pattern of inheritance studies. Then again, chronic periodontitis, also same thing. Polymorphisms associated with chronic periodontitis and pattern of inheritance studies. Then genetics in periodontitis is a manifestation of systemic disease. Then you have the diagnostic and therapeutic aspects, where you talk about the clinical utility of genetic testing in dentistry and salivomics, pharmacogenomics, nutrigenomics, epigenetic targets, and gene therapy. So what is periodontitis? We all know it's an inflammatory condition caused by microorganisms. But is it only by microorganisms? So it's also a host-mediated disease. And also the environmental factors influence it. And also it has a genetic component. So it is basically a complex disorder resulting in a group of diseases which have similar clinical features. They are periodontal diseases. So we have the 1997 model, which has demonstrating various contributing factors in the pathogenesis. This was given by Conman. So you can see here that the genetic risk factors, they affect the host immunoinflammatory response and connective tissue metabolism. When you write it as an answer, you have to draw the diagram and write about the genetic risk factors. If you draw the diagram itself, it will be more than enough, and just explain that it influences the host inflammatory response and the connective tissue and bone metabolism. Then we have the biological systems model, where again you can see here the genetic and the epigenetic level affecting the clinical phenotype. This has been given by Offenbacher. So you can mention this next. This is again the biological system model. That is, here it is actually the bacterial component, environmental factors, and the host genetic variation which are associated with the disease. So in this level A, they depict the biological mechanisms involved in the immunoinflammatory response and the bone and connective tissue metabolism. And in the section B, level B, they will show the clinical parameters and the biomarkers as a result of the interaction of factors in level A. That is a biological systems model. So this is the Chappell 2015 model. So yesterday, as Raisin sir had told, you can see that the variation that is coming in from clinical health to periodontitis. So here you can see that there is genetic risk factors absent, epigenetic effects not evident in clinical health. And as we move ahead towards periodontitis, you can see that there are genetic risk factors present and epigenetic effect evident. So you can mention this model also. Now we have been talking about epigenetics. Epigenetics, what is this epigenetics? So epigenetics, it is a phenotype. We told phenotype is a physical manifestation of the genotype. So phenotype, this is a phenotype resulting from changes without any alteration in the DNA sequence. So how does it happen? So this is genetics, where you use the DNA and you form the protein. Epigenetics is where other factors like inflammation, environment, bacteria, age, smoking, diet, all this can bring about some epigenetic modifications, such as histone modification, where there is acetylation of the core, methylation of DNA, microRNAs, which are RNAs which are involved in RNA silencing and post-transcriptional modification. All this happens in epigenetic modification. And there has been, so all this will alter how the body responds to the particular stimuli. Like, for example, in this article by Dr. Vamsi Lau et al., they have mentioned that due to the epigenetic influence, there is down regulation of signaling pathway with decreased activation of transcription factors. There is hypomethylation of IL-8, hypermethylation of COX-2. So basically, there is an alteration in the response of the body. And then there is this article by Jordansky, who have mentioned the influence of P. gingivalis on TLR2. Usually what happens is P. gingivalis induced activation of TLR2. It will induce the production of cytokines, chemokines, antimicrobial peptides, which will allow for the elimination of the pathogen. However, on long-term exposure to P. gingivalis, what happens is there is TLR2 promoter methylation in the gingival epithelial cell. That is an epigenetic transformation. So this TLR2 promoter hypermethylation will result in reduced TLR2 expression. So then what happens? There is reduction in the inflammatory mediator production and antimicrobial peptide. So there is the elimination of the pathogen gets affected. Then there is this concept of genetic dysbiosis. We have heard of polymicrobial synergy and dysbiosis. In genetic dysbiosis, it is said that group of dysbiotic microorganisms, they will contribute for the genes necessary for periodontal disease progression. Like, for example, all members of the red complex, they will exhibit an overrepresentation of RNA coding proteins, which are involved in the invasion of cells, and ferritin, which are linked to invasion of cells by P. gingivalis. One of the other gene is GROL gene. It is a virulence factor of aggregatibacter actinomycetin comitans. Then apart from this, the other part of genetic dysbiosis concept is that the susceptible host will have multiple potential disease modifying gene which will influence the bacterial selection, attachment and growth, and thereby alter the host immune response. So this, like for example, there is an association between the locus IQ42 and periodontopathogenic bacteria. And this brings to the concept of infectogenomics, which was first defined by Kellum and Weiss in 2006. It is the interaction between the host genetic variation and colonization by pathogenic microbes. Like for example, Nibali et al., they have reported that IL-6 hyperproducers, they are consistently associated with AA. And in this article by Gurjeet Kaur et al., they have mentioned many studies studying this infectogenomics, how the host receptors can alter the microbes. Like for example, in the studies by Schulz et al., CD14159TT genotype, they have reduced Prevotella intermedia detection. And here you can see in the study by Gong Y al, gingival overgrowth patient with CD14 260 CT plus TT, there is increased detection of PG, TF, TD, and red complex. So this is the concept of infectogenomics and pathogenesis of periodontal disease. So this is another article by Zheng Wei. There is a positive association between IL-10 ATA genotype and lower serum IL-10 levels. And also this is related to an increased count of AA in chronic periodontitis. So this comes, that is the part about the pathogenesis of periodontal disease models. Now let's come to the polymorphisms in relation to periodontal disease. So this classification, it has been taken from the article by Vijayalakshmi R. et al. And so this can be used as a mnemonic cram, that is the cytokine gene polymorphism, receptor and other gene polymorphism, where we have the Fc gamma, R2A, R3A, R3B polymorphism, then metabolism related gene polymorphism, HLA gene polymorphism, polymorphism in the innate immune receptors. Then there are some miscellaneous gene polymorphisms also. So now, like I said earlier in the flow chart, you need to tell what are the requirements in proving a disease or polymorphism association. So this is similar to your Koch's postulate, although it's not at all like Koch's postulate, but that requirement is the same. That is, the polymorphism should influence the gene product. The biases should be controlled and reported. Confounders should be factored into the analysis, and the affected gene product should be a part of disease etiology, or there should be some plausible biological explanation for it. So we have these various polymorphisms. You can learn this through you can use this table to learn the various polymorphism, IL-1 gene polymorphism, and you can see that there is association with chronic periodontitis, aggressive periodontitis and its association with IL-4, then chronic periodontitis, IL-6, then association with IL-10 with chronic periodontitis. So you have TNF alpha polymorphism and chronic periodontitis. There is no association with aggressive periodontitis. According to these authors, you have to mention the authors and the association or no association with which condition, chronic periodontitis or aggressive periodontitis. So these are some systematic reviews on cytokine polymorphism, IL-1 polymorphisms, then IL-4 polymorphism, then we have this IL-8 polymorphism. So you can learn this from this particular PPT itself, you can use and learn it, the various systematic reviews. Then the IL-10 polymorphism, TNF alpha polymorphisms. Then we have the receptor and other polymorphism like TLR2, TLR4 polymorphisms, which are not associated with chronic periodontitis, Fc gamma receptor polymorphism, estrogen receptor polymorphism, FMLP receptor polymorphism, the RAGE, CCR5, VDR, which is the vitamin D receptor. This has come as short note here in Manipal University, although I did not see this in the RGUHS paper. So then this continued receptor and other polymorphisms, we have CD14, HLA, MMPs, TGF, fibrinogen, all this. So you should learn the name of the author and whether it is associated with chronic periodontitis or aggressive periodontitis or not associated, whatever the studies have been done. So these are systematic reviews on Fc gamma receptor polymorphism, then vitamin D receptor polymorphisms, then systematic review on PRR polymorphisms. Then this is a HLA polymorphism systematic review. Here you can see that HLA A9, which we have learned in Carranza also, B15, they represent susceptibility factor for aggressive periodontitis. Then systematic review for MMP polymorphism. Now we have mentioned all the possible polymorphisms. Now if we are talking about, since it is genetics in periodontics, we come to aggressive periodontitis, although now it is not termed as aggressive periodontitis. But when you mention aggressive periodontitis, if you're writing a paper on genetics, you might have to mention it because of the previous studies that exist. So inheritance patterns, we will mention all the polymorphisms taken from the summary that is given earlier, if you are writing it as a separate short note. Otherwise, if it is a long question, you have already finished explaining the various polymorphisms associated with the disease, you don't have to repeat it. So then you have the inheritance patterns of localized aggressive periodontitis, X-linked dominant by Benjamin, Butler, Page, Van der Steen, then autosomal recessive by Forel, Jorgensen, then autosomal recessive again by Saxon and Bowman, autosomal dominant by Hodge. You can see the different patterns of genetic analysis also that has been done. Then recessive, then dominant by Mazarita in 1994. So these are the various pedigree analysis that has been done for aggressive periodontitis, the segregation analysis studies, linkage studies. Then there are genome-wide association studies which have shown the GLT6D1SNP associated with GWAS. Then even Andrew locus, it is associated with aggressive periodontitis. Then this is a twin study, but the thing is only one study has reported on the heritability in twins. So there is no clear conclusion based on the concordance rate of early onset periodontitis. Then chronic periodontitis, again, when you write on chronic periodontitis as a separate answer, you have to write the various polymorphisms based on that summary that was given earlier. But if you are writing it as a continuation to your answer, then you just go ahead with the various patterns for genetic analysis. You have the twin studies given by Mikulovic. His name is very important, Mikulovic et. The author's name is very important because he has they have done a lot of twin studies related to chronic periodontitis and genetics. So they have shown that monozygotic twins have a higher concordance of alveolar bone loss pattern than dizygotic twins. These are other twin studies for chronic periodontitis by Corey, LAER, et al. Then Mikulovic again in 1999. Then Mikulovic in 2000. Then 2000 study showed 50% heritability independent of smoking and dental services. Then we have the linkage analysis for chronic periodontitis, Tabita K. et al. In 2009. You remember the microsatellites? They are the short tandem repeats. Okay, they are associated with severe chronic periodontitis. So microsatellite marker in chromosome 19 associated with severe chronic periodontitis. Then we have the genome-wide studies where NIN, NPY, WNT5A has been associated with chronic periodontitis, severe chronic periodontitis, NCR2, 10P15, moderate chronic periodontitis. It is difficult to remember all the names. Try to remember a few at least. Then these are some of the genome-wide studies. This is for a Korean population by Hong et al. Then this is a genome-wide association study in the Japanese population where KCNQ5 on chromosome 6, Q13, GPR141, NME8 on chromosome. At least remember the name of the author, where the study has been done, and probably where that two of the suggestive loci for periodontitis in Japanese population. At least that much, if you can remember, that will be good. Then genetics in periodontitis is a manifestation of systemic disease. We can see that Papillon-Lefevre syndrome, Haim-Monk syndrome. These are autosomal recessive. Then Shediac-Higashi, then leukocyte adhesion deficiency type 2. All these are autosomal recessive, and they also have periodontitis associated with it. Whereas Ehlers-Danlos syndrome, cyclic neutropenia, these are autosomal dominant and have periodontitis associated with Marfan syndrome, Down syndrome, hyperimmunoglobulinemia E. These are the other syndromic conditions associated with periodontal disease. Now coming to the diagnostic and therapeutic aspect. So there is genetic testing in dentistry. Genetic testing for IL-1 genotype in dentistry began in 1997 with the introduction of the PST test. So to understand this was introduced to understand the patient's susceptibility to periodontitis. So periodontitis susceptibility trait test is a test which identifies the genetic predisposition of patient to periodontitis by detecting polymorphism in IL-1 gene. So basically IL-1A G, that is 4845, this is the location to T. Then IL-1B C3954T. Then there is a Perio gene which is for IL-1 beta B gene polymorphism. Okay, this is another genetic test. So this can come as Viva questions. Please remember what it is and what it studies. Then what is the clinical utility? Suppose you find that a person is genotype positive, so they obviously have an increased risk for severe disease. So you will go for more aggressive therapy, surgery, antimicrobial agents, and also more frequent recall and maintenance. So this DL et al. In 2015, they conducted this analysis of a large-scale study to show which was proposed to show that PST and Perio test they help to identify a subset of patients who experience fewer tooth extractions if provided with 2 annual preventive visits. That is, prediction of a genetic susceptibility allows, and the treatment for that, proper treatment for that, allows you to be less susceptible to tooth loss. Then there is this concept of salivomics. Salivomics is basically the study of diagnostic proteins like mRNA, metabolic compounds that are present in saliva. So you can find the microsatellite alterations of DNA, presence of IL-8, IL-1 beta, etc. using salivomics. And this will help to identify whether the person is likely to have periodontal disease, are more susceptible to periodontal disease. So how do you select patients for this genetic susceptibility testing? So patients with early signs of periodontal disease, periodontal maintenance patients, patients who are resistant to accepting treatment recommendations, new periodontal patients. Suppose you have biological family members of genotype positive patients, then patients with advanced periodontal disease, patients who are candidates for complex restorative procedures, then adult orthodontic patients. All these patients, you can do a genetic testing to identify whether they are susceptible for periodontitis, so that you can initiate therapy and control the disease process. Now, there is this concept of pharmacogenomics in the therapeutic aspect. That is, it is a study of the inherited variations in gene, which will control the body's response to the drugs. So this can be used to predict whether the patient will have a good or bad response to a drug or no response at all. Like you can have different combination. We want this particular drug not toxic and beneficial, but for each patient, the response will vary. So pharmacogenetics, it will evaluate how an individual's genetic makeup corresponds to the response to a particular medication. So we can tailor the medical treatment to the individual so that it increases the effectiveness and reduces the side effects. In pharmacogenomics, it combines pharmacogenetics with genomic studies. So you use large groups of patients to evaluate how the candidate drug will interact with the range of disease. And pharmacogenomics, which will increase the efficiency of the drug development process. Then we have this concept of nutrigenomics, which deals with the influence of nutrients on gene expression. So it also influences the genotype on the nutrient metabolism. See, so many nutrients, they will act as transcription factors, which in turn will influence the gene expression. Okay. So here for an example, there is selenium. It can modulate the MIT88 and TRIF dependent signaling pathways of TLRs, which leads to decreased inflammatory gene expression. This is an example of the importance of nutrigenomics in periodontal disease. And you remember the term epigenetics, methylation, etc., where it will alter the response of the body to the pathogen and any other stimuli. So you have epigenetic targets, that is histone deacetylase. It may be targeted by compounds like suberoyl anilide hydroxamic acid. So this will reduce the P. gingivalis and cytokine-induced CCL2. So it reduces the it controls or modulates the inflammatory response, thereby helping to protect the host. There is the BET proteins, DNMT. These are all the epigenetic targets. The epigenetic targets which will alter the DNA, like DNA acetylation, DNA methylation. All these are the functions of the what is happening in epigenetic modifications. So these compounds will affect these targets and thereby have a protective role on periodontal disease. From periodontal disease. And then there is this concept of gene therapy. So in gene therapy, in 1995, the first potential impact of gene therapy on dentistry was described. So basically, the goal of gene therapy, it is to transfer the DNA of interest into cells, thereby allowing the DNA to be synthesized in these cells and its proteins are expressed. Then there is another thing like a salivary gland of a mouse. When it is immunized using plasmid DNA encoding for P. gingivalis fimbrial gene, it produces like fimbrial protein locally in the salivary gland tissue. So what happens when you have this fimbrial gene produced in the salivary gland tissue? There is a production of salivary immunoglobulin A or IgA, which will act against the PG, which will sort of act like a protective agent against PG. Then also there is in rat models, and researchers have shown that immunization with genetically engineered streptococcus Gordoni vectors expressing P. gingivalis fimbrial antigen, they are sort of protective in rat models. And then there is the in vivo gene transfer for electroporation, by electroporation, which has been seen to affect the and have a positive effect on alveolar remodeling. This electroporation is when the DNA vector it is sort of forced into the cell using electrical impulse, that is electroporation, and has shown to have predictable alveolar bone remodeling. So then antimicrobial gene therapy is there for controlling the disease progression. So researchers have shown that when this host cells infected with beta defensin 2 gene via retroviral vector, there was an antimicrobial activity which enhanced the host antimicrobial defenses. These are the various potential uses of gene therapy in periodontitis. So in conclusion, when you're writing the conclusion, you can write what is periodontitis, a multifactorial disease, pathogenesis with a genetic implication, and various polymorphisms, etc., and utilization of this knowledge for diagnostic and therapeutic strategies. So this is a flow chart that you can go through, like what is periodontitis, theory of pathogenesis with a focus on genetics in pathogenesis, genetic polymorphisms associated with pathogenesis of periodontal disease, aggressive periodontitis, chronic periodontitis, all that. Then clinical significance of the knowledge of genetics, then role of genetics in diagnosis, treatment planning, and therapeutics, and give the conclusion. The next question was genetic marker. So you define the biomarker. Biomarker is a substance that could indicate a biological state and is an objective measure to evaluate the current and future disease activity. So now there is no description to this answer because everything was given in the previous answer. So what you should do when you get this question, you can write what is a biomarker, potential genetic marker. So in that you can write all the polymorphisms, genetic polymorphisms that are there, which can be potential genetic marker. They are not there because we have only genetic testing for the IL-1 gene polymorphisms. So then genetic markers in diagnosis, genetic testing in dentistry. Then you can write about salivomics, then clinical utility of genetic testing, again selection of patients for genetic testing in dentistry, and the conclusion. The next question was discuss the role of genetics in etiopathogenesis of aggressive periodontitis. You have to write, right now the definition only has changed, so we cannot say whether such a question will come for you, but this was there in the RGHS paper, so you can. Yes, ma'am. I'll finish in like 4 more minutes. Genetic polymorphisms implicated in pathogenesis of aggressive periodontitis. Then aggressive periodontitis, you can write the inheritance patterns. And from that table on infectogenomics, you can write about the infectogenomics in aggressive periodontitis and then give the conclusion. Then role of genetic polymorphism in initiation and progression of periodontal disease. Again, the flowchart is practically very similar. What is periodontitis? What are polymorphisms? Genetic polymorphisms implicated in etiopathogenesis of periodontal disease. Clinical significance of knowledge of genetic polymorphisms in periodontal disease. Role of genetics in diagnosis and therapeutics, and conclusion. Then again, the next question that had come was implications of genetics in periodontal disease. So the same thing. What is periodontitis? Theories of pathogenesis, genetic polymorphisms, all the aggressive, chronic, periodont. So you just have to learn one answer and you know all the answers. Then a few Viva questions that I had come through, that is xRNA in periodontics. xRNA, they have been proposed to play a role in variety of biological processes, intercellular communication, cell regulation, and recently a study by Urbanovics in 2019, they showed that 4 salivary xRNAs, it can be used to detect gingivitis. And then haplotype, it's a group of genes within an organism that was inherited together from a single parent. Then we have DNA positive and negative strands. So individual strand of DNA is referred to as positive sense if its nucleotide sequence is corresponding to the RNA transcript. You remember the transcription factor, transcription, translation, etc. I hope. So the DNA sequence that corresponds to that, it is the sense. And then the sense strand, then the antisense strand is the other strand of the double-stranded DNA molecule, and it is reverse to the or complementary to both the positive sense strand and the RNA. Then IL-1 polymorphism and smoking, you know this, Maguire and Nunn. IL-1 positive genotype, risk of tooth loss 2.7, then smoking 2.9, IL-1 positive plus smoking 7.7. This comes as a question. Then transcriptomics, it's a study of transcriptome, which is a complete set of RNA transcripts. So using microarray analysis. So its uses in periodontitis and periodontics. So Kepshulial, yesterday, Pauline ma'am had shown one diagram, one picture, a graph of a transcriptomic data. So they found that the current transcriptomic data, it does not align with the current classification of chronic and aggressive periodontitis, which is one of the reasons why we don't need aggressive periodontitis as a separate, or rather we needed a new classification process. Then Cho et al. In a transcriptomic study, they have shown that smoking related in DNA methylation patterns can lead to increased susceptibility to periodontitis. Then what is genomics? Genomics is a study of whole genomes of organisms and incorporate the elements from genetics. So thank you for your attention. And if you have any questions, my friend. Thank you very much. Jokes apart, of course, you can ask the questions. Sorry. Thank you.
Dr. Lavanya? Dr. Lavanya? Thank you, Dr. Lakshmi, for a wonderful
Thank you, ma'am.
For condensing such a complicated and a very dry topic. You have made your best efforts to make it interesting.
Thank you so much, ma'am.
And condense it as well for the ease of the students.
Thank you so much, ma'am.
Over to
I
Dr.
shall
Lavanya.
share my PPT with you, ma'am.
Yeah, you can share the PPT. There is one option of converting it to PDF that the technical team and the senior organizers are planning it over. So
yes, ma'am. Yes, ma'am.
Dr. Lavanya?
Yes, madam. Yes, madam.
Yeah, please take over.
Yeah. So also, madam emphasized on the role of epigenetics, genomics, and the salivomics. These are relatively new, and they play a major role in deriving the newer techniques for aiming at specific therapeutic models. And also she also mentioned about the patient selection for genetic susceptibility testing, who would be the ideal participants to get tested, like the patients who are new to the periodontal disease or who have high risk of genetic influence, etc. And also we learned about pharmacogenomics and the nutrigenomics, how micronutrients play a major role in the process of mutations or polymorphisms. And also at the end, we learned about gene therapy, which has been researched a lot in our field, because we are aiming at that particular gene to act upon, and which will help us to cure the disease. With this few, at the end, we also spoke about periodontal vaccination, which is a dream. And I hope we achieve something like the COVID vaccination, which happened within a short period of time, hopefully in the near future. With all these few words, I like to end this. And ma'am, that was a wonderful presentation once again. And I think
Thank you very
one of
much.
the most tough topics, which was very much simplified for the students to understand. I think even it was to the level of the BDS students. If they would have sat also, they would have understood this chapter very easily. So
Thank you very much, ma'am.
I think it was of great use to our postgraduate students who are going to appear for this examination. And wish you all the best, students, for the examinations. And uh, I request you to accept the token of appreciation for presenting this lecture, madam.
Thank you very much, ma'am. Thank you very much. And all the best to all the students. Do well in your exam. Don't be stressed. You have time to prepare, and prepare well and make a flow chart and learn well. All the best.
Thank you, madam. Thank you.
Thank you very much. And thank you very much, BAP, for giving me this opportunity. And I also thank my teachers for guiding me throughout, and my department here in Manipal, who have always supported me. Thank you very much.
With this, we come to the end of the first session for today.
The next
Fauziya,
lecture
ma'am?
in series is by Dr. Sapna.
Yeah.
Dr. Sapna, are you there?
Yes, Dr. Fauzia, I'm here. Am I audible?
Yes, you are audible. Dr. Lavanya?
Yes, madam, I'm here. I'm here. The second topic for today's session is periodontal medicine. And good morning once again to one and all. Madam, it gives me immense pleasure to introduce you to the crowd. Dr. Sapna completed her BDS in the year 1999 and post-graduation in 2006 from College of Dental Sciences, Davangere. She is currently working as reader and PG guide in RV Dental College, Bangalore. To her credit, she has 50 publications and 12 presentations to her credit in both national and international journals and platforms. She was a part of the team in organizing pre-conference course for the ISP National Conference in the year 2010. She has she has also actively involved in organizing Oral Hygiene Day celebrations from BAP in the year 2013, and was one of the resource persons for the scientific sessions. She is also involved in various academic activities, being part of panel discussions and as guest speaker. Ma'am, I welcome you and I kindly request you to take over.
Thank you, Dr. Lavanya, for that wonderful introduction.
Thank you, ma'am.
I will start sharing my screen.
Yeah.
Is the screen visible?
Not it, ma'am.
Is it visible now?
No, ma'am.
No.
No.
1 minute.
Yes, ma'am. Now it is
Is
visible.
it visible now?
Yes, ma'am. Yes, ma'am.
Okay, thank you one and all. And a very good morning to all dear students, all the organizing team of Perio Panorama. First, I would like to thank BAP and the organizing team of Perio Panorama for giving me this opportunity. And also, I would like to thank my HOD, my colleagues, and my postgraduate students for supporting me throughout this presentation. So, today the topic for my presentation is periodontal medicine. So, what is this periodontal medicine? Medicine. When we talk about medicine, we usually think that it may be related to some analgesics, antibiotics, or any other pharmacological agents that are used. But are we talking about this medicine? No. Here we are talking about the medicinal part of periodontal treatment. So, what is the influence of medicinal part on the periodontium, and what is the influence of periodontal implications on medicinal part? So, what is the link between this oral health and the overall health? How this one system will affect the other system, and what is the evidence? So, that is what we are going to discuss today in this presentation. So, I'll be covering this presentation under the following headings, where I'll be discussing individual systems with the link to the periodontitis. So, to begin with, what is the definition of periodontal medicine? It is defined as a rapidly emerging branch of periodontology, focusing on the wealth of new data, establishing a strong relationship between periodontal health or disease and systemic disease or health. If you look at this definition, it says that it is two-way relationship. That is periodontal health having an effect on the systemic health and systemic health having an effect on the periodontal health. However, the recent research has focused more on the role of periodontitis in affecting the systemic health, and this has led to the emergence of new branch of periodontology, that is periodontal medicine. And this whole presentation today will be concentrating on this relationship between periodontitis and other systemic diseases. So, is this periodontal medicine a concept new? Are we recently dealing with that periodontitis can have an effect on the systemic disease? No, it is not something new. It was proposed way long back in 1890 by W.D. Miller, who presented a paper titled The Microorganisms of the Human Mouth, the Local and General Diseases which are caused by them. Later, in 1900, William Hunter, he proposed that oral sepsis is a cause of disease. And also, he condemned the procedures where the decayed tooth were restored. And he said that they are following a septic dentistry, and he attacked on the conservative dentistry. He said instead of restoring them, they should be extracted because they are the source of infection. This was followed by Frank Billings in 1911, where he spoke about focal infection and focus of infection, and he advocated removal of all foci of infection. Martin H. Fisher, he advocated radical dental treatments like full mouth extractions, and he said this is the dentist's dilemma. However, the tides turned, and the Dental Cosmos, a journal in the field of dentistry, wrote an editorial where they called for return to constructive treatment rather than destructive treatment. Cecil in 1938, he observed that there was no improvement in rheumatoid patients' condition even after full mouth extractions. And Williams and Burkett in 1951, they said that there is no evidence of improvement in the systemic conditions if you are going to extract all the infected teeth. However, they also mentioned that you should try to eliminate the focus of infection just for overall hygiene measure. So then there came a workshop of the American Academy of Dental Medicine, and the topic of discussion in that workshop was focal infection with relation to dental medicine. In that workshop, periodontal group came to a consensus that there is a direct relationship between periodontal condition and an associated systemic problem. However, endodontic group said that there was no systemic contraindications for endodontic treatment of teeth. All these discussions led to a more constructive type of treatment rather than radical extraction of all teeth. Journal of American Dental Association in 1952 stated that focal theory has fallen out of favor. This they said because they observed that when foci of infection was eliminated from many patients who were suffering from systemic conditions, there was no relief from these systemic conditions. And they also observed that focus of infection was present even when there were systemic conditions. Sorry, just a second. Even when there was foci of infection, there were no systemic conditions in many of the patients. So they said focal theory is not supporting the present scenario. Then came the time of focal infection revisited in the year 1994, when again the focal infection theory started gaining popularity. However, this time it was backed by the evidence which was not there in the previous time. So in this article, they mentioned that the oral microorganisms can contribute to systemic diseases through metastatic infection, metastatic injury, and also metastatic inflammation. So can you see this phylogenetic like tree? It's like a circle wheel. So this is given in the reference is given below, where it is a clinical trial registry where the trials are registered. Almost 57 systemic diseases are hypothesized to be related to periodontal disease, and they are registered under this trial. Can you imagine 57 systemic conditions have been hypothesized to be related to periodontal disease? If you can see this photo, you can see almost all systems related to periodontal disease: immune system, skin and connective tissue, nervous system, digestive system, bacterial infections, nutrition, musculoskeletal. So you name the system, it has been related to periodontal disease. So now we know that oral cavity and systemic disorders, they are interlinked. So now what is the link? Whether this link is only association or is there any causal relationship? And how do we decide what is the link and should we agree to this or not? So whether we agree or not, it should be based on the evidence, with a good, strong evidence based on the hierarchical evidence. So let's move on to the individual systemic conditions which are related to the periodontal disease. The first one is cardiovascular system. So the cardiovascular conditions that are related to periodontal disease are atherosclerosis, coronary heart disease, myocardial infarction, and cerebrovascular accident or stroke. So basically, whenever I'm discussing about any particular system, I'll be discussing about the mechanism through which periodontitis and these systems are interrelated, if there are any proposed mechanisms, and the evidence. But because of the time constraint, I'll be focusing more on the biological mechanism and less on the evidence, because anyway, the PPTs will be shared with you and you can have a look at the evidence. So what is this atherosclerosis? It's a progressive disease which involves large to medium sized muscular and the large elastic arteries. And they finally lead to coronary heart diseases, myocardial and cerebral infarctions. So what happens during this atherosclerosis? Inflammation is the key factor in developing of atherosclerosis. Low density lipoproteins, they start accumulating in the arterial wall, and a signal is given to the endothelial cells, and these endothelial cells, they produce various chemicals like monocyte chemoattractant proteins and other endothelial molecules which will attract these monocytes or macrophages, and these monocytes, they enter into the vessel wall, and there they start engulfing on the low density lipoproteins and they become foam cells. And these foam cells, from here there starts a formation of fatty streak, which is the earliest stage of atherosclerotic plaque, and it starts growing, and it enlarges, and it leads to narrowing of the aortic lumen. And at the same time, this plaque is also covered by a fibrous cap. But due to various reasons, it may be inflammation or any other trigger, the cap surrounding the plaque can get destabilized, and can it obstruct the blood flow and can trigger a heart attack. This is what happens in atherosclerosis and myocardial infarction. Now coming to what happens in periodontal disease, how this is linked to coronary heart disease. Platelet aggregation is a very important step during atherosclerosis and thrombus formation. And the microorganisms that are associated with periodontal disease, like P. gingivalis and also S. sanguinis, they have a protein called platelet aggregation associated protein, which we call it as PAAP. They help in platelet aggregation. And also these bacteria, either they directly attach to the bacteria or through their bacterial products, they will activate these platelets. And this is the factor for thromboembolic events. The next mechanism is these bacteria, the periodontal bacteria, they enter into the endothelial cells and macrophages, and they enhance the foam cell formation. And also they produce increased levels of cytokines, which can lead to cellular dysfunction and atherosclerosis. Now, these increased cytokines, they will also lead to cholesterol accumulation, smooth muscle proliferation, and thickening of the vessel wall. And they also inhibit fibrinolysis, they facilitate coagulation and thrombosis. So all these events will lead to atherosclerosis formation, then later maybe thromboembolic events. So this is the mechanism by which periodontitis is linked to the coronary artery diseases. So this is a model proposed by Offenbaker in 1999, where he said that because of the periodontal infection, production of reactive oxygen species that leads to atherogenic LDL formation that can enter systemic circulation, leading to atherosclerosis. Another model by Fong et al., where he showed that periodontitis is a risk factor for coronary heart disease. The microbes, periodontal microbes, either through direct invasion, they cause injury, or through indirect way, that is producing cytokines and endotoxins, they can trigger increased levels of LDL, leading to atherosclerosis formation, and also later increased platelet aggregation, they stimulate thrombosis, then followed by ischemia. This is the mechanism. And there are various proofs available in the literature to confirm the role of periodontal infections in cardiovascular disease. For example, periodontal bacteria can be present in the systemic tissues, they can be present in the affected tissues, they can also induce atherosclerosis in animal models. And when you insert a non-invasive mode of bacteria, they will reduce. These are the proofs available to prove the role of periodontal infections in cardiovascular disease. Coming to the evidence, these are the studies which shows that systemic inflammatory mediators like C-reactive protein, interleukin 6, and etc. are increased in case of chronic periodontitis. Fibrinogen again is increased in chronic periodontitis, and it decreases following periodontal therapy. These are the epidemiological studies like cohort study, case control studies, which shows that there is an association between myocardial infarction or cardiovascular disease and periodontitis. This is the periodontal intervention. If you do the treatment, whether it will reduce the risk for coronary artery disease or not. They said that when you do the treatment either with antibiotics or without antibiotics, the systemic biomarkers reduce in count. However, till now, none of the intervention studies have shown that primary outcomes like incident of cardiovascular disease and cerebral stroke is affected by periodontal treatment. So again, periodontal treatment can reduce almost all the systemic biomarkers and leading to decreased incidence of cardiovascular disease. This is the Bradford Hill criteria, and it shows that cardiovascular disease and periodontitis fulfills almost all criteria, and thereby you can say that there is a definite causal association between periodontitis and cardiovascular disease. To summarize, periodontal infection can influence formation and progression of atheromatous plaque, and there is also an association between these two conditions. Periodontal therapy reduces serum inflammatory mediators and also, but however, there is no reduction in the risk for cardiovascular disease. So we can say there's a great chemistry and there's a great friendship between heart and teeth, and they forever they sing "Ye dosti hum nahi todenge." So at this time I would like to tell all my students that please refer to this particular article. This is a very beautiful article written by Beck et al. In 2019, where he has spoken about the 100 years of periodontal medicine. And he has mainly concentrated on cardiovascular disease, adverse pregnancy outcome, and pregnancy. And you get a flow chart and what has happened, the significant studies that has happened from 1900 to 2020. Please refer to this article. These are some of the systematic reviews and meta-analysis in this. Next is infective endocarditis. I will not go much in detail about this. Infective endocarditis is a microbial infection of the endothelial lining of the heart. Mainly bacteria are responsible for this. However, periodontal pathogens rarely cause infective endocarditis. These are some of the conditions which predispose a person to develop infective endocarditis. And some of the dental procedures which have been shown to cause bacteremia that can lead to infective endocarditis. If you look at this table, say that extraction can cause bacteremia in 10 to 100%, periodontal surgery in 36 to 88%, and so on and so forth. And dental prophylaxis is about 0 to 40%, and endodontic therapy 0 to 20%. However, the role of dental procedures as a cause of infective endocarditis is very controversial. So this is the antibiotic regimen. I will not discuss in detail amoxicillin or ampicillin, and if they are allergic, you go to clindamycin or azithromycin. So now, when should you give antibiotic prophylaxis? In 2007 guidelines, they said that antibiotic prophylaxis has to be given during all dental procedures that involves manipulation of the gingival tissue and if you are doing any endodontic therapy. Same thing was followed in 2015 also. However, when the guidelines were proposed in 2016, they mentioned that antibiotic prophylaxis is actually not routinely recommended for dental procedures. Why? Because the risk of developing bacteremia comes more from poor oral hygiene and dental disease than from dental procedures. So it is prudent to emphasize more on oral disease prevention than the routine use of antibiotics. Okay. Next condition is diabetes and periodontitis. We know that there is a two-way relationship between diabetes and periodontitis, each affecting the other system. So but I will be concentrating on the influence of periodontitis on diabetes. But before I go ahead, I'll just show you a flow chart where during diabetes, there will be hyperglycemia, there will be shift in the gingival flora and also dyslipidemia. This can lead to increased cytokine levels, increased stress and immune dysfunction. So there will be Rankel and OPG interaction, oxidative stress leading to enhanced bone and connective tissue destruction, leading to exacerbation of the periodontitis. Now, what is the effect of periodontitis on diabetes? First thing is periodontitis worsens the glycemic control. Second, it increases the diabetic complications. It is a predictor for future ischemic heart disease. And the interesting part is it may also play a role in the incidence of new cases of type 2 diabetes. For example, if patient is not having diabetes, but if patient is suffering from periodontitis, maybe in future he will develop diabetes mellitus because of the presence of periodontitis and also gestational diabetes. However, till now, the role of periodontitis on type 1 diabetes is not established. And it is also seen that in healthy periodontitis patients without diabetes, there is moderate hyperglycemia. So what is the mechanism? What is the effect of periodontitis on diabetes? In untreated periodontitis, there are circulating bacteria and bacterial antigens, elevated levels of cytokines leading to insulin resistance and exacerbation of diabetes. At the same time, there are elevated counts of HbA1c levels leading to increased diabetic complications. So this we already discussed. There's a significant increase in HbA1c levels and also dyslipidemia, and there is chances of increased risk of developing type 2 diabetes mellitus. So these are the studies which showed that there is an association between periodontitis and diabetes, and also there was increased levels of systemic biomarkers in patients with systemic periodontitis which were related to diabetes. Now, what is the influence of periodontal treatment on the glycemic control? When patient undergoes periodontal treatment, there is reduced levels of bacteria and bacterial antigens, and at the same time reduced cytokine levels. These two will in turn lead to improvement in the insulin resistance, thereby improving the HbA1c levels. So improved HbA1c levels, that means less diabetes complications. So periodontal treatment has modest effect on the glycemic control, and this effect on HbA1c is seen approximately 3 to 4 months after the periodontal therapy. So these are the studies showing the effect of periodontal treatment on the diabetes. It shows that periodontal treatment may alter adiponectin, CRP levels, but however, in another study, they showed that there was no reduction in the CRP levels. Again, this is a flow chart given in the same article that I was mentioning. The first study on showing the influence of periodontal therapy on insulin was done in 1960, and the latest is in 2018, where they did intensive periodontal therapy and they found that there was significant improvement in the HbA1c levels. These are the systematic reviews and meta-analysis in this link. Next, coming to the periodontal disease and respiratory system. So, lower respiratory infections are the third commonest cause of mortality, and COPD is the sixth leading cause of mortality. There is an anatomic connection between the oral cavity and lungs, and they also share microflora. So, periodontal diseases may influence the course of respiratory diseases. So, now what is the role of bacteria in respiratory infections? They have seen that respiratory pathogens are present in the dental plaque, and they can enter the lower bronchi through aspiration and can cause infection in the lung fluids. So now, what is the mechanism? Sorry. If you see this photo, you see there are 4 mechanisms. First one is P. gingivalis produces enzymes. Because of these enzymes, the receptors are altered, and to these receptors, respiratory pathogens like H. influenzae can get attached, colonize, and can invade the respiratory system. Next mechanism is the salivary pellicle is removed from the mucosal layer by P. gingivalis. This leads to, again, exposure of the receptors. You can see these blue colored pictures, they are receptors. They are receptors for H. influenzae. They get attached and they enter the respiratory system. Third mechanism is, usually these H. influenzae is covered by a salivary pellicle, which prevents its attachment to the receptors. However, P. gingivalis produces enzymes which will remove these pellicles so that H. influenzae is exposed and can get attached to the receptors. Fourth mechanism is production of cytokines, which then enter saliva and they upgrade the receptors for H. influenzae. So again, there is an interaction between H. influenzae and the receptors, and H. influenzae can invade the respiratory system. So this is the mechanism. So now coming to the individual conditions, basically pneumonia and COPD. In pneumonia, you have an inflammation because of the microbial infection. 2 types, community acquired and hospital acquired. and we are more concerned about the hospital-acquired pneumonia. So a study was done where they said that respiratory pathogens were seen in 65% of patients in ICU compared to 16% of outpatients with respiratory pathogens. So now what is the mechanism? How these respiratory pathogens from the oral cavity can enter the lower respiratory tract? First is through aspiration of the oropharyngeal contents. Second, inhalation of infectious aerosols. Third, spread of infection from contiguous sites, and hematogenous spread. Once they enter, they cause infection in the lower respiratory tract and they lead to pneumonia. So, however, what is the role of periodontal treatment? So, they said that when you do periodontal treatment, it can reduce the risk of pneumonia. However, they found very little evidence that it reduces the length of hospital stay or mortality caused by pneumonia. Next is chronic obstructive pulmonary disease. What happens here? There's a chronic obstruction to airflow resulting in excess production of sputum, and then it leads to chronic bronchitis and emphysema. So what happens? How this oral disease or periodontal disease is related to this? So there is a hypothesis called as vicious circle hypothesis revised. Okay, in this, there are various initiating factors for COPD, like smoking, childhood respiratory disease, and altered microbiome. Altered microbiome, it can also be seen in case of periodontal disease. When these factors are there, there is impaired mucociliary clearance. And this in turn leads to altered microbiome. So it can cause inflammation. And at the same time, various products are produced which will cause injury to the airway. So all these in turn will lead to COPD. Even in periodontal conditions, what happens? There is microbial dysbiosis leading to exacerbation of this vicious cycle, and also release of cytokines, which then release MMPs that leads to exacerbation of the COPD. So this is the link between periodontal disease and COPD. So to summarize, oral microbiome is an important determinant of lung microbiome. However, certain areas are unexplored still, like if you change the oral microbiome, whether it will have any effect on the lung microbiome, and if you do any periodontal therapy, will it improve the lung health? These research is still in infancy stage. So these are the systematic reviews and meta-analysis. Next condition is periodontitis and adverse pregnancy outcomes. What are the adverse pregnancy outcomes? Preterm low birth weight, preeclampsia, miscarriage, and intrauterine growth retardation that are related to periodontal disease. So, Offenbacher was the first person to identify maternal periodontitis as a risk factor for preterm low birth weight infants. And also he linked this to the severity of periodontitis. So now, what is the proposed model that links periodontitis to adverse pregnancy outcome? So this is the flow chart. There are three mechanisms by which periodontitis is related to adverse pregnancy outcome. First is metastatic infection. Second is metastatic injury. And third is metastatic inflammation. In periodontitis, there is bacterial infection, which is local in nature. And also because of bacterial infection, there is injury. And now these bacteria, they enter the fetoplacental unit. Once they enter fetoplacental unit, they cause infection in placenta, amniotic fluid, etc. And also because of the infection, there is inflammation. And in inflammation will lead to production of prostaglandins. Once there is production of prostaglandins, membrane ruptures and there is uterus contraction and there is preterm birth. And also because of the inflammation, there is nutrition supply is stopped or reduced to the fetus, leading to impaired organ development and leading to miscarriage or mortality or morbidity or long-term disability. And also because of the inflammation, there is imbalanced angiogenic and anti-angiogenic factors. At the same time, these bacterial byproducts, they also stimulate maternal liver to produce various acute phase reactants like CRP and fibrinogen. All these together will cause imbalance in the angiogenic and anti-angiogenic factors, leading to preeclampsia, low birth weight, and preterm birth. Various microorganisms, periodontal pathogens, are associated with these adverse pregnancy outcomes. Important ones are Porphyromonas gingivalis, Burgella species, and Fusobacterium nucleatum. So these are the studies done in this field. Here they show that there is an association between preeclampsia and maternal periodontitis, and the association were statistically significant in few other studies. These are the studies where they did non-surgical periodontal intervention and they saw if they had any influence on the adverse pregnancy outcome. The study by Offenbacher in 2006 said that it did influence and reduce the adverse pregnancy outcome. The second the study, it was done by our very own Sir Dr. Faizuddin Sir and Dr. Fauzia Taranum, where they compared treatment group and control group, and they said that treatment resulted in reduced incidence of adverse pregnancy outcomes. However, there are various studies which have shown that there is no effect of periodontal therapy on the adverse pregnancy outcomes. So, the question is, why is periodontal therapy is not having any effect on the adverse pregnancy outcome? Sorry. Because usually periodontal therapy is done during second trimester. And by this time, the bacterial byproducts would have already reached the fetoplacental unit and they may have induced irreversible metastatic injury. So it is prudent and it is more meaningful to restore the maternal periodontal health during preconception period to improve the pregnancy outcomes rather than to improve it during pregnancy. So again, this is the flow chart given in that article. Please refer this. So these are the systematic reviews and meta-analysis. Next condition is periodontitis and metabolic syndrome. So what is this metabolic syndrome? It has various components like visceral obesity, insulin resistance, hypertension, high triglycerides, and low HDL cholesterol. If a person has at least 3 of these components, he is said to be suffering from metabolic syndrome. So one of the components of this metabolic syndrome is obesity. And obesity and periodontitis, they share various common risk factors like genetics, socioeconomic position, lifestyle, and aging. So now, what is the link between obesity and periodontitis? In obesity, you have increased adipokines, appetite hormones are elevated, cytokines are elevated, reactive oxygen species, all these are elevated, leading to inflammation, insulin resistance, and hyperglycemia. And we know the link between the diabetes and periodontitis leading to periodontitis. And in obesity, gingival microcirculation is also affected. There is hyposalivation, there is stress, all leading to periodontitis. Now, what is the role of periodontitis in obesity? Again, in periodontitis, appetite hormones are increased, leading to increased food intake, leading to obesity. Periodontitis inflammation, so you have increased cytokines, leading to insulin resistance and obesity. In periodontitis, there may be tooth loss and mobility. What happens when there is mobility and tooth loss? Patient prefers soft, high-calorie diet, leading to obesity. Periodontitis may lead to aesthetic problem, halitosis, leading to limiting the physical activity and social activity, again leading to obesity. So these are certain mechanisms by which periodontitis can be linked to obesity. Now coming to the other metabolic components like dyslipidemia, etc., how they are related to periodontitis. Periodontitis is said to be associated with decreased high-density lipoproteins and increased low-density lipoproteins. Two-way relationship is present between periodontitis and dyslipidemia. And periodontal infection is shown to promote lipolysis and upregulation of the triglycerides. However, dyslipidemia has shown to increase the pro-inflammatory state, predisposing a person to periodontitis. Next component is diabetes. We know already what is the link. So next one is hypertension. And we have already discussed how periodontal disease can lead to atherogenosis and thrombus formation that itself can lead to increased risk of hypertension. So these are the studies which said that there is an association between metabolic syndrome and periodontitis. This is link between hyperlipidemia and periodontitis, link between hyperglycemia and periodontitis, all showing that there is an association. Hypertension and periodontitis, and obesity and periodontitis. And this is the treatment response after periodontal treatment. They said after periodontal treatment, there was improved healing in obese individuals. So it is shown that periodontal therapy improves the metabolic components. And weight loss also will have a beneficial effect on the periodontal parameters. And this link, it not only has clinical implication, but it also has public health implications. So these are the systematic reviews and meta-analysis. Next condition is periodontitis and rheumatoid arthritis. So what is rheumatoid arthritis? It's a chronic inflammatory and autoimmune diseases affecting the joints. The first biological link between these two conditions was proposed by Neiderman in the year 1982. But that is not it. Way back during Hippocrates' time, it is said that Hippocrates cured arthritis by pulling out the infected teeth. So it dates back to so long. So now what is the link between this rheumatoid arthritis and periodontitis? They said that periodontal bacteria are associated with chronic inflammatory host response and they trigger rheumatoid arthritis. And they also found periodontal pathogens in the synovium of rheumatoid arthritis patients. So now the mechanism, this is called as two-hit model given by Golub et al. In 2006. So what happens initially, if you see here, number one, there is periodontal infection. Periodontal infection leads to periodontal inflammation. And also in infection, there is Porphyromonas gingivalis. And Porphyromonas gingivalis has shown to citrullinize the bacterial as well as host peptides. Once there is citrulination of the peptides, antibodies are produced against this. Antibodies are present in the periodontium, antibodies are present in the alveolar bone, and antibodies, they also reach various joints and bones. So next stage, if there is any infection or inflammation in the joints of other body parts, already there are antibodies present to against this citrulination. And this infection again leads to citrulination of the peptides. Now already there are antibodies produced. Now there is exaggerated antibody production leading to severe rheumatoid arthritis in periodontitis patients. So these are the studies linking rheumatoid arthritis with periodontal disease, and they found that there is an association between these two conditions. And also they found that there was an association between anti-rheumatic drug treatment and periodontal parameters. And these are the studies which shows the role of Porphyromonas gingivalis in rheumatoid arthritis. So this is the effect of periodontal treatment on rheumatoid arthritis. They said that after treatment, there was improvement in the markers of disease activity in these patients. However, all these studies were of a short duration and with low sample size. Next, effect of treatment for rheumatoid arthritis on periodontal parameter. Mainly for the treatment of rheumatoid arthritis, they give host-modulating agents, anti-cytokine factors. So when anti-cytokine factors are given, it reduces cytokine levels, and also it's said that it reduces periodontal inflammation. However, they also mentioned that periodontal disease may hamper treatment response in rheumatoid arthritis patients. So there is an association between these two conditions, but no causal relationship. However, if proven, periodontal therapy can be an inexpensive and non-pharmaceutical way of improving rheumatoid arthritis. These are the systematic reviews and meta-analysis. Next condition is periodontitis and cancer. So periodontitis is related to various cancers. It may be pancreatic cancer, gastric cancer, breast cancer, lung cancer, many cancers. So now what is the link? The first plausible mechanistic link is in periodontitis, you have inflammation, and inflammation leads to oxidative stress. This stress leads to DNA mutation and reduced DNA repair, so leading to cancer. And at the same time, free radicals, they also again stimulate inflammatory cells, which again will cause more inflammation. More. Okay, I'll try to finish fast. Okay, this is the link. And also they said that periodontal pathogenic organisms cause damage to the cells and cause mutation. Next is they increase the level of CRP, causing low-grade inflammation, leading to precancerous lesions and cancers. So, studies have shown that there is an association between periodontal disease and overall cancer risk, especially in relation to cancers of lung and upper digestive tract. So, these are the various cancers and their association. Most of it have positive association, except for colorectal cancer, which is found to have no association with periodontitis. These are the reviews. Next is Alzheimer's disease. In Alzheimer's disease, we'll tell you what happens. Cytokines and bacteria are released. They are there in the peripheral compartment. They also enter the brain through this blood-brain barrier. Once they enter there, they lead to formation of amyloid B, and cytokines are released. This amyloid B leads to neuronal degeneration. In advanced case, it leads to neural damage and irreversible brain injury. This is the link between Alzheimer's and periodontitis. So, not only it may be there is a biological plausibility between these two conditions, but they also said along with bacteria, even herpes virus could also act independently or synergistically in pathogenesis of Alzheimer's. Next condition is very relevant today, COVID-19 and periodontal disease. I'm not going about the coronavirus aspect, it is very in many patients it causes mild symptoms, but in certain patients it requires hospitalization and few people even succumb to this coronavirus. So now what is the mechanism? Mechanism is that when you have periodontitis, these bacteria are aspirated inside and they express angiotensin converting enzyme 2, and this is a receptor for COVID virus. So to this receptor, this COVID virus gets attached and lead to increased cytokine levels. The second mechanism is periodontal pocket could act as a viral reservoir. And the third mechanism is there's a strong Th17 response in severe periodontitis patients that can lead to cytokine storm in COVID-19, leading to increased complications and many times death of the patient. So when the question is asked for 100 marks, you have to include this and along with a few more conditions which I'm not discussing because of the time constraints. They are systemic lupus erythematosus, liver disease, chronic kidney disease, hypertension, irritable bowel syndrome, erectile dysfunction, and osteoporosis. You can include these conditions also. To conclude, periodontal medicine is a term used to describe how periodontal infection or inflammation can impact extraoral health. And I've already mentioned there are more than 50 conditions that are hypothesized to be linked to periodontitis. So what is the significance of this branch of periodontology? It has a great public health significance. Why? Because periodontitis is both preventable as well as treatable. So if you can prevent and treat periodontitis, you can actually prevent and improve the prognosis of various systemic diseases and conditions. With you here, I know that life is much more fun. Don't you agree with this? If you have a good periodontal health, the life will be much more fun. Thank you. And these are the questions that were asked in the RGHS exams. I have written the flow chart for individual questions. When you get the PPT, so I think you will be able to look at these flow charts. Thank you for your patient hearing, and all the best to all the students. Do well. Thank you.
Thank you, Dr. Sapna. That was a real wonderful and a detailed presentation regarding periodontal medicine.
Thank you so much, ma'am.
Yeah, thank you, ma'am. There were few questions. One was like the effect of periodontal therapy on cardiovascular disease.
Yes, ma'am. What evidence has said that it has an effect on the secondary outcomes, like secondary outcomes in the way the systemic biomarkers. It reduces CRP levels, it reduces platelets, it reduces hypertension, it reduces various fibrinogen, WBCs. But the primary outcome, like incidence of cardiovascular disease or incidence of stroke, that is not proven yet. So only the secondary outcomes have been proved using the periodontal intervention.
Ma'am, thank you so much. The other one is like the role of HP1AC on periodontal disease.
Sorry, ma'am, I didn't get your question.
Role of HP1AC
Okay, ma'am.
on periodontal disease.
Okay, ma'am. Whenever there is diabetes which is uncontrolled, there is increase in HbA1c levels. Whenever there is increased HbA1c levels, there is increased oxidative stress, and also there is increased levels of cytokines, and they in turn lead to more of destruction, and there is more of interaction between advanced glycation end products and receptors for advanced glycation end products. These two will lead to increased periodontal destruction and exacerbation of the periodontitis.
Thank you so much, ma'am.
I hope answered this question.
Yes, ma'am. Thank you so much. With this, we come to the conclusion and the take-home message for this session on periodontal medicine is that it's a branch of periodontology on which mainly deals on establishing the relationship between the periodontal health and systemic diseases. And we have, Madam has listed the various commonly seen systemic diseases that are encountered and the relationship between periodontal disease and the systemic diseases. So most of the times, other than diabetes mellitus, which has shown established a relationship, the others are not yet established. We are still in a hypothetical stage. So the effects of periodontitis on diabetes is that it worsens the glycemic control and increases the severity of the diabetic complications and also increases the death of the person, or mainly due to the cardiovascular or the renal complications that are encountered. And also, in case of untreated patients, especially with diabetes, who are suffering from diabetes mellitus, it usually leads to exacerbation of diabetes levels. And also, with periodontal treatment or intervention in most of the systemic diseases, the bacterial load is decreased, and because of which the interrelated mechanisms are interrupted. And with these few words, I try to end this session. And I thank Dr. Sapna for that wonderful session and hope all our students have been helped and they would have understood the periodontal medicine topic in detail, and which will help them to write better answers in their examination.
Thank you, Dr. Lavanya.
Thank you, ma'am. I request Dr. Sapna to accept the token of appreciation for presenting this session.
Thank you so much, ma'am.
Thank
you for giving me this opportunity to present.
Thank you.
Thank you so much.
Thank you.
Thank you, Dr. Lavanya, for consenting to moderate this session for today's program. We are grateful for the time and effort that you have contributed to this program. Now I request the technical team to sign the certificate of appreciation for Dr. Lavanya.
Thank you so much, ma'am. Thank you, ma'am. I thank the organizing team and also the BAP for giving me this opportunity to moderate 2 sessions. I'm very much indebted to the society. Thank you so much.
Thank you. The speaker for the next session is Dr. Vijay Raghavan. This session will be moderated by Dr. Nisha. Now I hand over to Dr. Nisha.
Thank you, Dr. Fausia. I hope I am audible.
Yes, you're audible, Dr. Nisha.
Yeah. Thank you, Dr. Fausia, and good morning, everyone. I thank Bangalore Academy of Periodontology and the organizing team of Perio Panorama 2021 for giving me this opportunity. I'm privileged to introduce our next speaker for the day, Dr. Vijay Raghava. Dr. Vijay completed his BDS from Oxford Dental College in 2001 and his MDS in Periodontics from A.B. Shetty Memorial Institute of Dental Sciences, Mangalore in the year 2007. He started his teaching career in Sri Rajiv Gandhi College of Dental Sciences, Bangalore, and presently he is professor in Department of Periodontics in the same institute. Dr. Vijay has numerous publications to his credit in both national as well as international journals. So with great pleasure, I invite Dr. Vijay to speak on the next topic, risk factors and risk assessment in periodontal disease. Over to you, Dr. Vijay. Dr. Vijay.
I had spoken to Dr. Vijay. He said he is connecting.
Okay.
Maybe some technical issues. Really sorry for the inconvenience.
So from, I think, till the time Dr. Vijay connects, I'll just give a brief intro on the topic. So from an exam point of view for postgraduates, this is a very relevant topic, especially after its inclusion. Before also we know that smoking and diabetes as a risk factor were very important, but especially since its inclusion in the latest 2017 classification, we know that a few of the risk factors have been included as grade modifiers, right, in periodontal disease. So I'm sure Dr. Vijay will be giving an insight on all these topics. He'll be talking on the various risk factors as well as the various measures of risk assessment. So we'll wait for Dr. Vijay to connect.
No, see, you should take care. The internet connectivity needs to be good. Yeah. Please log in. We are waiting. We are waiting for you, please. Yeah. I think there are some connectivity issues with Dr. Vijay. A few more minutes, he will log in.
Yeah.
The postgraduate students should show some interest. They have to post some questions. If there are no questions, then the discussion cannot happen in an appropriate manner. Some doubts, some information taken from the lecture, and if you have questions on it, requesting postgraduate students to post some questions which will help a discussion between the moderator and the speaker.
Yes, we'll take up the queries from the chat box. So dear postgraduates, please come prepared for the topics so that you will have some doubts at the end of the session. Though the PPTs and all will be shared, but still you can clarify the doubts from speaker themselves. So that will be of great use for the postgraduates.
Dr. Nisha will be moderating two lectures. Dr. Vijay Raghavan's lecture, that is on risk factors, risk assessment and periodontal disease. Another lecture followed by this is by Dr. Lalit Vivekanand. The topic is advances in periodontal diagnosis. Both the lectures will be moderated by Dr. Nisha.
Thank you.
Please do not add any more messages stating add our number to the WhatsApp group. I have made it very clear for those who missed it. I have announced that there is no such WhatsApp group. It was a misunderstanding created by one of the postgraduate students only, who asked whether there is a WhatsApp group, and the others thought that there is a WhatsApp group. So there is no such WhatsApp group being created. Yes, an attempt will be made to pass on the material to the postgraduates, either as PDF files or as PPTs, after discussion with the speakers and the technical team. How it will be done will be confirmed by tomorrow.
Any message from Dr. Vijay? Dr. Fausia? Dr. Fausia, do we have any communication from Dr. Vijay?
Ma'am, he says he's got some connectivity issue. He said he logged in and then the internet got disconnected. I spoke to him, ma'am.
Oh, okay. Okay, Dr. Fausia.
Yes, ma'am.
I think Dr. Vijay has logged in.
We regret the inconvenience being caused. This is the inherent issues with the online program. I hope all the participants understand this. Dr. Vijay is trying to log in. We will just give him a couple of minutes. Dr. Vijay? Sorry, Dr. Nisha.
No problem, no problem. I can see Dr. Vijay logging in. Yes, I think he is connected. Dr. Vijay?
Sometimes these are all connectivity.
Yes, ma'am. Yes, ma'am.
Yes, okay. Dr. Vijay, I finished your intro. So I invite you to take over the virtual.
Can you see
podium.
the?
Yes, yes.
Can
we
you
can
see the screen, ma'am?
Yes, we can see the
Yes,
screen.
Dr. Vijay, and please hide the dialog box that is appearing on the screen.
Which one? Hide, ma'am?
Below
hide.
your that PPT slide, there is one sharing screen thing so that you can
hide. That's a dialog
hide
box.
it.
Yeah.
Yes,
Thank you.
yes.
Okay.
Yes, Dr. Vijay.
Yeah.
Over to you.
Fine, ma'am.
Yes, yes, you can.
Right, ma'am. Sorry, extremely sorry for the inconvenience. Yeah. Extremely sorry for the inconvenience. Now, today's topic of presentation is risk factors and risk assessment in periodontal diseases. So I'm Dr. K. Vijayaraghavan, Professor, Department of Periodontics, Sri Rajiv Gandhi College of Dental Sciences. So today I'll be dealing with, my contents of the presentation is, I'll be talking about the introduction, definitions, basic terminologies, what are the different risk elements. I'll be talking about the clinical risk assessment for periodontal diseases, models for periodontal risk assessment, summary, conclusion, and references. So, basically, risk assessment involves identifying elements that either may predispose patients to developing periodontal disease or may influence the progression of the disease that already exists. So, evaluation of the factors contributing to the risk, patient's education, concerning the risk, and when appropriate, suitable intervention strategies should be implemented. So risk is an event which will occur in future or probability that an individual develops a given disease or experiences a change in the health status during a specified interval of time. Okay. What is risk assessment? So according to the American Academy of Periodontology in 2008, risk assessment is defined, please mark my words, risk assessment is defined as the process by which qualitative or quantitative assessments are made of the likelihood for adverse events to occur as a result of exposure to specific health hazards or by the absence of beneficial influences. So basically, when a question is asked to you, they might ask you as a long essay, 100 marks question also. It can come as, what enumerate the various risk factors that is associated with periodontitis and write about the clinical risk assessment and add a note on risk assessment tools. That is what I'm going to deal with today. Okay. So risk can be identified in terms as risk factors, risk determinants, risk indicators, and risk predictors. So basically, we have to know these terminologies. So what is a risk factor? A risk factor may be environmental, behavioral, or biological factors that, when present, increase the likelihood that the individual will get the disease. Okay, and it has been well established in both cross-sectional and longitudinal studies. Whereas risk determinants are those risk factors that cannot be modified. Next is risk indicators. These risk indicators are the probable risk factors that have been identified in cross-sectional studies but not confirmed through longitudinal studies. Whereas risk predictors or markers, they are although associated with the increased risk of the disease, but they do not cause the disease on their own. Okay, so these are the four important things. Okay, first is the risk factors, then is the risk determinants, then is risk indicators, and finally it is risk predictors or markers. So some of the examples of each: first is the risk factors, that is tobacco smoking, diabetes, pathogenic bacteria, and microbial tooth deposits. Risk determinants, examples are genetic factors, age, gender, socioeconomic status, and stress. Risk indicators are HIV and AIDS, osteoporosis, infrequent dental visits, and risk markers are previous history of periodontal diseases and bleeding on probing. Okay, so I'm going to deal with each. I'm just give a gist of it. I'm not going to elaborate on each. Okay, in your answer, you have to elaborate on each topic. Okay, so what is a conceptual model? There is by James Kroll in the year 2017. He has given a conceptual model where the periodontal status is evaluated basically by bacterial biofilm, immune and inflammatory factors, host and genetic factors, and all the other factors, that is the social determinants, familial determinants, and the individual determinants will revolve around these factors. Okay, and this is not only about risk factors, these factors also play a very important role in determining the risk assessment tools also. Okay, so the first and foremost, we are not going in detail, we're just telling what are the factors. Okay, so basically tobacco smoking is a well established risk factor. There is a direct relationship exists between smoking as well as the prevalence of the periodontal disease. Okay, and as you all know, products from the tobacco smoke, mainly the acrolein, acetaldehyde, nicotine, carbon monoxide, hydrogen cyanide, benzopyrene, etc., they all result in functional alterations in chemotaxis, phagocytosis, and oxidative burst. And they also delay apoptosis. There is increased production of pro-inflammatory cytokines, mainly the interleukins, 1 alpha, 1 beta, and TNF alpha. There is enhanced osteoclastic activity. There is reduction in the IgA levels, and hence there is suppression of the antibody response, which is very much essential for phagocytosis as well as bacterial killing. The IgG2 levels are also reduced in smokers. There is elevated levels of prostaglandins, MMP8, and neutrophil elastase. They've all been demonstrated in smokers. And further, this nicotine adversely affects the fibroblast function, and thus it further suppresses the osteoblast proliferation. Thus, there is the normal reparative and regenerative process of the periodontium is altered, which further leads to disease condition of the periodontium. So there is a strong correlation between smoking and periodontitis. Hence, it is considered as a very, very important risk factor for periodontitis. Okay, there are a lot of studies, one by Grossi et al. In 1995 have shown that smokers were at greater risk for experiencing severe bone loss when compared to that of the non-smokers. And studies have also shown that there is a dose response effect, increase in consumption of cigarettes a day. There is more likely that the individual, or say more likely that individual will develop periodontitis. Okay, and basically, and further studies have shown that patients who refrained from smoking for the entire study period had significant reduction in probing depth when compared with that of the smokers. So there is definitely a reduction in non-smokers. And systematic review given by Patel et al. In 2012, it has shown that smoking negatively affects bone regeneration, Further, Fabio in 2018 in a systematic review have also told that there is increased breakdown of periodontal tissues through impairment of immunological as well as vascular mechanisms. The second important risk factor here is the diabetes mellitus. Okay, there is a clear risk factor for periodontitis. Okay, and lot of epidemiological data demonstrate that the prevalence and severity of periodontitis are significantly higher in patients with type 2 or type 1 diabetes mellitus than those without diabetes. So the level of diabetic control is an important variable in this relationship. So basically, in diabetes, it is a bidirectional relationship. But now I'm going to talk about only the relationship of diabetes and periodontium. How diabetes affects the periodontium. Basically, the function of immune cells, including the neutrophils, monocytes, as well as macrophages, is often altered in cases of poorly controlled diabetes. The neutrophil adherence, chemotaxis, and phagocytosis are altered. As a result, there is increased destruction of the periodontal tissues. Further, the neutrophil function is diminished in diabetes, but there is an hyper-responsive monocyte macrophage phenotype, which further increases the production of pro-inflammatory cytokines. And diabetic patients with HbA1c levels more than 8% produce higher levels of interleukin and TNF alpha than the non-diabetic individuals. There is altered collagen activity, where the diabetic individuals are more susceptible to degradation of collagen due to increased production of collagenase. There is inhibition of osteoblastic proliferation. There is altered wound healing. Further, there is alteration in the microvascular integrity also. And you have to explain about the increased production of advanced glycation end products. You have to talk about the interaction between age-RAGE, that is the receptors that is present in the monocytes and macrophages. They interact with the advanced glycation end products, which will further result in increased cytokine production, and it causes endothelial cell dysfunction and vascular proliferation. Okay. Further, there is impairment of bone formation and repair. There is alteration in the periodontal microflora, where there is increase in the putative gram-negative microorganisms are more in number. And there is increased insulin resistance, which can be due to obesity or due to the production of cytokines or due to the reduced production of adiponectin, which further results in the periodontal breakdown. Okay, so these are important salient features of how diabetes plays a very important role in periodontitis. You can elaborate upon these things in your answer. Okay. Further, a lot of studies, PIMA Indians and NANS 1 survey, NANS 2 survey has shown that, there is a strong correlation. It's been established, a two-way relationship between periodontal disease and elevated glucose level in a large population. You can talk about complications of diabetes also. You can write about the sixth complication, that is periodontitis given by low. These are all can be included in your answer. Okay. Then these are important studies again by Grossi, Taylor, which shows that the incidence of periodontitis is reduced in better controlled diabetics when compared to the poorly controlled diabetes. And patients with the type 2 diabetes mellitus have a greater susceptibility for more severe periodontal disease when compared to non-diabetic individuals. Okay. So the third important point is the microbial dental plaque, or the bacteria. Okay. See, the ultimate risk factor for an infectious disease is a causative agent of the disease. Without any agent, no disease will take place, no matter what other risk factors the subject may possess. Okay. There is a strong correlation between bacteria. Okay. It is the principal villain in bringing about periodontitis. Okay. So in a World Workshop in 1996, they have evaluated and finally they've concluded that the 3 specific bacteria have been identified, like Aggregator-Bacter actinomycetum comitans, Porphyromonas gingivalis, and Tandrilla forsythia have been included as the principal offenders in bringing about periodontitis. Okay, so here you have to talk about what is the importance of plaque, how there is pellicle formation, how there is adhesion, how there is co-aggregation happening, and further you have to talk about the concave appearance. You have to talk about the different complexes given by Hafaji and Sokransky. You have to talk about the different subgingival microbial complexes. You talk about the green complex, the orange complex, the red complex, how it will come. What are the importance of these red complex? Why it is called as a climax community? Then you have to add a note on the plaque hypothesis. Both quantity and quality of the plaque, how it is determined. You have to add a note on Sokransky's criteria, why cost postulates failed in determining the association between plaque and periodontitis. Okay, then you have to add about the influence of lipopolysaccharides. You have to talk about leukotoxins. You have to talk about the virulence factors, how virulence factors are capable of adhering and invading the periodontal tissue. You have to talk about how the host cell is penetrated, how it will induce apoptosis also we have to speak. Okay, then add a note on biofilm. What are the special properties of biofilm which makes the plaque resistant, and then what is the difference between plaque within the biofilm and plaque in its planktonic state, and what are the special properties of biofilm, like special enzymes or multi-drug resistant pumps or quorum sensing, which makes the plaque more resistant to antibiotic as well as to the treatment modalities. Okay, so again, various studies have shown not only about the red complex and AA comitants, but also suggestive of the involvement of Proteola intermedia or Campylobacter rectus and other important microorganisms, which can be suggested as an important risk factor for progressive periodontitis. And further, presence of calculus also serves as a reservoir for bacterial plaque as a risk factor for periodontitis. Okay, now the next we'll go ahead with the apart from it we have to talk about the second one, that is the risk determinants. As I told you earlier, risk determinants are those risk factors that cannot be modified. The first and the foremost is the genetic factors. I don't want to elaborate on this. There's various studies like twin studies, linkage studies, segregation studies, association studies. In the morning, Dr. Lakshmi would have thoroughly explained regarding the association between genetics and periodontitis. So you have to elaborate mainly on the twin studies, and you have to talk about the studies done by Conman et al. And where there is a specific interleukin-1 genotype association with severe chronic periodontitis. Okay, and also you have to talk about the different polymorphisms, like tumor necrosis factor alpha gene polymorphism, vitamin D gene polymorphism, NAT2 gene, N-acetyltransferase 2 gene polymorphism, how it is associated. Okay, Kinan et al. In 2003 also suggested that interleukin-1 gene alterations may be a valid marker for periodontitis. Okay, the various systematic reviews, which has shown that there is a positive correlation between interleukin-1 genotype and periodontitis. There is an increase in the progression of the periodontitis in those patients who have shown interleukin-1 genotype positive. Okay. Now coming to the tooth factors as risk, basically there are various tooth factors like furcation involvement, an association between enamel projections and furcation involvement. It has been hypothesized that enamel in these locations prevents the attachment of the connective tissue, thus making the area more susceptible to periodontal breakdown. Any root abnormalities like palatogingival groove, in maxillary incisors have been shown to be associated with periodontal breakdown. Overhanging restorations might contribute to the periodontitis. Pulpal diseases especially might contribute, particularly when there is pre-existing periodontitis in patients. Okay, and tooth malposition, malalignment, crowding, migration, or tipping of the tooth distal to edentulous areas have also been implicated in the loss of periodontal support. In addition to it, any labial or lingual positioning of the teeth has been correlated with gingival recession, and the risk is even stronger in malposed teeth with suboptimal oral hygiene. Okay. Now, age. Okay, age, the prevalence and severity of the disease increases with age. It has been suggested that periodontal disease is not an inevitable consequence of aging process, but due to increased intake of medications, due to decreased immunofunction, due to altered nutritional status, age plays a very important role. Okay, and age is also been included as one of the parameters in in the risk assessment, this thing also, and certain physiological changes in periodontium occur with age, although these changes are not alone responsible for bringing about the periodontal destruction. Okay, so certain studies have shown, when compared to that of the 25-year-old, the attachment loss and bone loss, the annual rate of bone loss and attachment loss is said to be 0.28 mm when compared to the 25-year-old, which is only 0.07 mm. Okay. Gender plays a role in periodontal disease. It is, a lot of studies have shown that males have more loss of attachment than females, and they have poor oral hygiene than the females, as evidenced by higher levels of plaque in calculus. And these are some of the studies. Stress, very important. Manifest in periodontium through behavioral changes like increased smoking and poorer oral hygiene. And various models have also been described, how stress plays a very important role, where it will activate the HPA axis, that is hypothalamus pituitary and autonomic nervous system axis, where activation of CNS will help in release of ACTH, that is adrenocortisol hormones, which further depresses the immunity. There is reduction in IgA levels, there is reduction in IgG levels and TMN alteration, which in turn results in increased production of interleukins and MMPs and results in periodontal breakdown. And the stress can again activate the neurotransmitters like epinephrine, norepinephrine, bradykinin, and other neurotransmitters, which in turn increases the prostaglandins and protease levels, which in turn causes periodontal diseases. Further, the psychosocial stress can bring about behavioral change, which in turn can cause poor oral hygiene, poor compliance, which in turn causes bacterial infection and further results in periodontal disease. There can be overeating, which in turn results in increased cortisol production. Depressed immunity will further result in periodontal disease. The psychosocial stress again can, the patients can, the smoking can be increased in these patients who are undergoing stress, and smoking in turn can cause periodontal disease. So these three important models you have to illustrate whenever you're writing the answer. Now, various systematic reviews have shown that there is a positive correlation between stress and psychological factors. These are some of the studies which have shown that the salivary cortisol and beta endorphins were statistically significantly associated with tooth loss and clinical parameters of the periodontal disease. Socioeconomic status. Gingivitis periodontitis can be related to lower socioeconomic status. So this can mostly be attributed to the decreased dental awareness and decreased frequency of dental visits. And a lot of studies have been shown, where the prevalence of gingival bleeding, loss of attachment was found to be more with the decrease in the socio-economic level. Okay. Again, the oral conditions, the prevention strategies is different in a well-educated individual when compared to that of the non-educated individuals. Next is the risk indicators. So we have spoken about risk factors. We have spoken about risk determinants. The third is risk indicators. Risk indicators are those risk factors, that has been demonstrated only with the cross-sectional studies and not longitudinal studies. So first and foremost is HIV and AIDS. The AIDS adversely affects the host resistance to the presence of bacterial plaque, and thus it can predispose some individuals to increase in the incidence or severity of the periodontal disease. So mainly the periodontal conditions infected with HIV are the linear gingival erythema, necrotizing ulcerative gingivitis, necrotizing ulcerative periodontitis, as well as chronic periodontitis. So you can elaborate on each, what are the important salient clinical features and how there is periodontal breakdown. And see, studies have shown that, whenever the CD4 counts are less than 400 and whenever the CD4 count is less than 200, there is a positive correlation between the severity of periodontal disease and the level of immunosuppression. Next is osteoporosis, which comes under the risk indicators. It results in loss of bone mineral density throughout the body, including loss in the maxilla as well as the mandible. Although studies in animal models indicate that osteoporosis does not initiate periodontitis, but definitely there's a lot of evidence has been shown that it might aggravate the periodontal disease progression. So there is a strong interrelationship between the oral bone loss and systemic bone loss. And studies have also shown that the risk of periodontitis decreases in patients who are receiving estrogen replacement therapy. Okay. So next is the infrequent dental visits. Again, it can be attributed to various factors like socioeconomic status or poor patient complaints or negligence of the patient. Also, there is an increased risk for severe periodontitis in patients who are not visited the dentist for 3 or more years. And more loss of attachment and bone loss is seen in individuals who did not seek dental care compared with those who did over a 6-year period. Okay. These are some of the studies. So before going to the risk markers, and even certain neutrophil dysfunctions are also included as the risk indicators, like the Chidiac-Higashi syndrome, lazy leukocyte syndrome, Papillon-Lefebvre syndrome, Down syndrome, systemic lupus erythematosus, etc. Okay. And finally, we have the risk markers. Okay. Risk markers are those that are strongly associated with the disease, but they themselves do not cause the disease. 2 important factors are there. One is the previous history of periodontal disease. So patients with the previous history of periodontal disease is a good clinical predictor of risk for future diseases. Okay. Then we have bleeding on probing. It is a very good, best clinical indicator of gingival inflammation. So bleeding on probing with increasing pocket depth can be an excellent predictor of future loss of attachment. And lack of bleeding on probing does appear to serve as an excellent indicator of periodontal health. So various studies have shown that BOP is more likely to lead to future attachment loss. It is said that 90% of the pockets with bleeding on probing will be associated with future attachment loss. About 3-year, 4-year study period was conducted. Okay, so next we will talk about the clinical risk assessment for periodontitis. So basically, one of the what is risk assessment? As I told you earlier, it is a qualitative or quantitative assessment that are made of the likelihood for adverse events to occur as a result of exposure to specific health hazards or by absence of beneficial influences. Okay, why there is a need for risk assessment? Okay, first and foremost, we can create a patient profile. Two, we can characterize the patients. Further, we can set up the priorities. We can we can plan, or we can create a strategy for effective management. And finally, effective intervention can also be planned, if we have a proper risk assessment model or a risk assessment tool. Okay, so there is something called as the Hills criteria. Okay, Hills criteria means, see, it was Bradford Hill in the year 1965. He has given certain criteria to prove the association between the risk factor and the disease. What he said was, for the risk factor to bring about the disease, there should be a strong strength of association. There should be a strong correlation between the risk factor and the disease. Two, consistency. It should be repeatedly observed in different persons, in different places, in different circumstances and different times. Three is specificity, whether it affects a specific age group or a specific population or a specific site. There should be temporal consistency, that is temporality should be there. That is, if a particular situation or a particular occupation or a particular environment will promote the infection or the disease process. Most important, it should be biologically gradient, that is biological plausibility should be there. That is, some sort of evidence should be present in the literature. There should be a dose response effect, that is quantitative measurement also plays a very important role. As the dose of exposure increases, the risk of the disease also should increase. There should be coherence. Coherence means, the cause and effect interpretation of the data should not conflict with the generally known facts of the natural history and biology of the disease. Okay, then experimental, that is, does the preventive action taken on the basis of the cause effect relationship, does it alter the frequency of the disease outcome? And there should be considerations of an alternate explanations in the form of analogy. Okay, so these criteria should be met for it to call as a risk factor. Okay, the second one is the Beck's principle. Okay, the Beck's principle is, it involves three important parameters. One is the identification. Okay, first and foremost is the identification. That is, the risk, it is considered as the risk factor if the exposure has occurred prior to the onset of the disease. Okay, and the exposure or the factor should be consistent with the current understanding of the disease process, and it should be well established in longitudinal studies. Then these risk factors are subjected to various multivariable risk assessment models. Okay, thus the assessment, you can assess the exposure and you can prospectively identify the people as high risk category, low risk category, and medium risk category. And finally, once the identification is done, then with the help of risk model, you are able to assess the categories. And once the category is assessed, then you can target, with the help of precision medicine, you can target that particular high risk category, by modulating or by altering the modifying factors, by altering the intervention strategies. You can actually target the high risk category and lower the risk of the disease in these individuals. Okay, so these are the important salient points which you have to talk in the clinical risk assessment. That is, you have to talk about Hill's criteria, you have to talk about Beck's principle, then you have to talk about the different assessments. See, one of the problems with risk assessment in periodontal disease is that the periodontal disease are multifactorial, and the assessment should be therefore to be carried out at multiple levels. So you can consider 4 levels here. There can be patient level assessment, tooth level assessment, mouth level assessment, and site level assessment. The patient level assessment can be performed at the initial examination. So what you have to do in initial examination, a familial history to be taken, any hereditary factors are there to be taken, a detailed history of any early loss of tooth in the family, medical history in the form of osteoporosis, diabetes mellitus, etc. Present dental history, how well the patient is motivated, so you can improve the compliance of the patient. Okay, social history in the form of smoking, etc. Habits like bruxism, etc. can be assessed in the patient level assessment, which is to be performed at the initial examination. The second one is the mouth level assessment, to be performed at the initial examination and during the post initial therapy. Here, basically occlusal examination can be carried out, examination of levels of oral hygiene, examination of levels of plaque retentive factors, levels of recession, any gingival inflammation and depth of the pocket can be assessed, and examination of attachment loss relative to age can also be assessed. The third is the tooth level assessment. The tooth level assessment is to be performed during the post initial therapy, during the definitive therapy, and during the maintenance therapy. Here again, individual tooth mobility can be assessed. Any drifting or pathological tooth migration can be assessed. Periodic radiographic evaluation, whether there is a bone gain or bone loss following the treatment, can be assessed. Furcation lesions can be assessed, and any soft tissue contours, in presence of subgingival calculus, overhanging margins and restorations, anatomy of the tooth embrasures and contact points, if there is variations, can be assessed in the tooth level. And finally, site level assessment to be performed after the post definitive therapy and during maintenance. Any bleeding on probing, exudation, attachment levels, or any other anatomic factors like enamel pearls, root grooves, etc., can be assessed during this site level assessment. Okay, so these are the 4 important ones, that is the patient level assessment, mouth level assessment, tooth level assessment, and site level assessment. Okay. Now, lastly, the last aspect is the risk assessment tools. We'll just go through certain risk assessment tools. Okay. The first and foremost is, I'm just briefly telling you about this risk assessment tools. The first and foremost is the oral health information suite. Okay. This system basically, this is a very important system. It quantifies not only the current periodontal disease state, but it also quantifies the risk for the future disease. Okay, and a diagnosis is made by attributing all the related queries regarding the periodontal disease. A diagnosis is made, and a risk score and a disease score is calculated. Okay. Further, once the treatment is performed, the risk score and the disease score is again analyzed and evaluated. And if it is not satisfactory, again, the treatment can be modified. The second important is the periodontal risk calculator. Okay, it is a computer-based risk assessment tool which will take into consideration 9 important factors. That is the age of the patient, smoking history, diabetes, history of periodontal surgery, pocket depth, furcation involvement, restoration or calculus, radiographic bone height, and vertical bone lesions. These 9 factors are considered, and the presence or absence of these factors will give either the scoring of low risk or highest risk. Okay, a 5-point risk scale is used to balance the sensitivity of the risk assessment with the time and the cost required to obtain it. Okay. The third important is the periodontal assessment tool that is given by Lang and Tonetti in the year 2003. This is an hexagonal risk assessment diagram. It considers 6 important components. One is the bleeding on probing to be measured percentage wise. 2 is the prevalence of residual pocket that is greater than 5 mm. 3 is loss of teeth, from a total of 28 teeth, how many tooth is lost over a period of time. 4 is the periodontal support, loss of periodontal support in relation to patient's age. 5th one is systemic and genetic conditions, that is diabetes, etc. And the 6th one is the environmental factors such as cigarette smoking to be determined on the number of cigarettes smoked by the individual. So the 6 parameters are taken and the patients are divided as low PRA, moderate PRA or high PRA. That is, low PRA means you can see here, there is an hexagonal, basically, in the hexagon there you can see in the center of the polygon there is a low risk category, in the periphery is the high risk category, and in between this high risk, low risk and high risk is the medium risk category. So when do you call it as low PRA? Low PRA means when the patient has all parameters within the low risk categories, at most one parameter is present in the moderate risk category. Moderate PRA means, patients has 2 parameters within the moderate risk categories, but at most one parameter in the high risk category. Whereas the high PRA is patient has at least 2 parameters in the high risk category. Okay, this was modified by Chandra. What he has done is he has incorporated 2 new categories. Apart from the 4 categories which was involved in the original PRA model, diabetes is included as a new category, and other background characteristics like stress and socioeconomic status and infrequent dental visits, etc., has been incorporated as a new category. Okay, again, based upon this, he has evaluated patients as low risk, moderate risk, and high risk PRA model. Okay, and there is one more given by Trombili where 5 categories are involved. Smoking status, diabetes, number of pockets, bleeding on probing, bone loss and age ratio. So a sum score of 0 to 2 is referred as low risk. From 3 to 5 is medium low risk. 6 to 8 is medium risk. 9 to 14 is medium high risk. And 15 to 14 is high risk. Okay. Apart from it, there are other thing. One is called as dento risk. The dento risk, again, it incorporates basically 20 factors. The factors are divided as systemic factors and local predictors and systemic predictors. There is something called as Cronin-Stassen-Betz chasm scale. Here the B stands for BMI, E stands for ethnicity, D stands for diabetes, S stands for stress, C stands for college, that is, the education level, H stands for hygiene, A stands for age, S stands for smoker, and M stands for male. So each scoring is given. If most of the things are positive, then a very high risk category, the patients are involved in high risk category. And lastly, it is the risk assessment based individualized treatment called as RABIT, where the risk assessment is done as a part of the initial diagnosis, then the risk assessment to be carried out in the recall schedules also. So recall appointments driven by different risk factors can be combined in single recall appointments in order to enhance the efficiency and patient compliance. And lastly, the genetic test is also available to determine the periodontal disease risk. The retrospective evidence that the genetic testing for specific interleukin genotype may give an indication of increased susceptibility of tooth loss in periodontal maintenance patients. Okay, so These are some of the questions asked in the previous RGHS question papers. They have spoken about risk assessment in periodontal disease, risk factors in periodontal disease, write a small note on risk assessment models, and separately, smoking can be asked as a risk factor, diabetes mellitus can be asked as a risk factor, role of genetics, role of microbes, etc. So to summarize, today we have spoken about what is risk, what is the definition of risk factors. So what are risk factors? What are risk determinants? What are risk indicators? And what are risk markers? Then we have spoken about the clinical risk assessment, a small note on Hill's criteria, a small note on Beck's criteria. Then we have spoken about the individual tooth assessment, whole mouth assessment, patient assessment, as well as the site assessment. Then we have spoken about the various periodontal risk assessment tools, starting from oral hygiene information suite, then periodontal risk calculator, Langen-Turnitin hexagonal risk diagram. Then we have spoken about modification given by Vishwachandra. Then we have spoken about the Unify risk assessment tool. Then we have spoken about the AAP periodontal assessment tool. Then we have spoken about the Stronin chassis Betz chasm assessment tool. And finally, we have spoken about rabbit and the genetic test. Okay. So thank you very much. So I would, in the outset, I would like to thank all the organizing committee, and I would like to thank my mentor, my philosopher, Dr. Sarita Narayan Joshi ma'am, for providing me an opportunity to speak. Thank you very much. Thank you, ma'am. And extremely sorry for the delay. I sincerely apologize.
Thank you for that very clear and simple presentation, Dr. Vijay. I really appreciate the way you condensed such a vast topic, and you concentrated more on like how to approach a question on risk factors and risk assessment. That is really appreciated. Here, actually, we have one question in the chat box, that is, in case of former smoker, whether the assessment of prognosis will depend on the number of years the patient has left the habit. That is
yes, question. Definitely yes, and the amount of cigarettes smoked by the patient previously also plays a very important role. Okay, and the number of years also.
Yes.
Whether when he was a current smoker, how many cigarettes was he smoking? That also plays a very important role. Okay, for the cessation of the symptoms, and the number of years also.
Yes, I would like to add on that also. There is one recent one survey which is published in JOP in the year 2018, one NAN survey, where they observed that with each additional year of since quitting smoking, there was a significant reduction in the odds ratio by around 3.9%. So okay. That also shows that definitely have an effect on the prognosis, yeah. Right? So another thing is, I would like you to add some, throw some light on the clinical relevance of including risk factors in our recent 2017 classification, especially in this era of precision medicine, right?
Yeah, ma'am, that is what, the risk factors, as I told, plays a very, very important role. Okay. Yes. So primarily, we have to identify the risk factor. Yes. Okay. Once the risk factor is identified, then we have to assess it, whether that particular risk factor, does it play a very important role in bringing about the disease process? So you can classify it as high, moderate, or low risk groups. Okay, if you feel that that particular risk factor will bring about the disease process in the individual, then we can modify. In the present AAP classification, the risk factors, they are used as grade modifiers. Especially smoking and diabetes mellitus have been included as grade modifiers, and precisely if we can control these risk factors, it could be critical for the long-term stability of the periodontium.
Okay.
So it can be, we can include it as a grade modifier where we can modify accordingly and we can plan the intervention and the treatment so that we can provide a long-term stability of the periodontium.
Yes, good, very nice. So another question we have is can trauma from occlusion be considered as a risk?
Yeah, trauma from occlusion can also be considered as a risk factor, but again, a lot of studies have shown. Finally, I feel they have concluded that there should be presence of the pre-existing gingivitis or periodontitis for trauma from occlusion. To it can actually cannot bring about the disease process, but definitely it can increase the progression of the disease in the pre-existing gingivitis or periodontitis.
Thank you, Dr. Vijay, for that excellent presentation and the clarification of all the queries. Please accept a token of appreciation, Dr. Vijay.
Yes. Thank you, ma'am.
Yeah, thank you, Dr. Vijay.
Thank you. I once again apologize for the delay. I'm extremely sorry.
No problem for that. We are still in that our time frame.
Yeah.
Okay. Thank you, Dr. Vijay.
Okay, ma'am. Thank you very much. Thank you. Thank you, ma'am. Thank you.
So before we wind up today's morning session, we have one more topic, that is advances in periodontal diagnosis. So with great pleasure, I introduce our next speaker, Dr. Lalit Vivekananda. He is presently working as Associate Professor in M.R. Ambedkar Dental College and Hospital. He completed his BDS from the same college, M.R. Ambedkar Dental College and Hospital, MDS from BS Dental College, Bangalore, and also obtained his PhD from M.R. Ambedkar Dental College under RGHS. He holds several degrees in oral implantology, that is Masters from Oral Implantology from York University, USA, Fellowship in Oral Implantology from Cambridge, UK, this to name a few. Dr. Lalit has lectured in various platforms in India and abroad. He also has several publications to his credit. So over to you, Dr. Lalit. Dr. Lalit.
Thank you, Dr. Nisha.
Yeah.
Am I audible?
Yes, yes, you are audible, Dr. Lalit. Yes, we can see.
Good morning, everyone. Is the screen visible?
Yes, yes, Dr. Lalit, you can continue.
Okay, yeah. Thanks a lot. Good morning, everyone. It's great to be a part of the Perio Panorama 2021, the first online event of all the Perio Panoramas been conducted over the years. I'm really glad to be a part of it and discuss the topic on advanced periodontal diagnosis for today. I'll be discussing the topic under the following headings. This will also be the outline for your the main questions. What I've done is I have segregated the topic and condensed it so that we can stick on to the time limit. The Viva part, whatever the questions might be asked, I have indicated as a red marking in throughout my presentation. So whatever comes in red as the points or as the title will all be your Viva questions. Now, coming back to the topic, periodontitis is a multifactorial disease. As a clinician, we have to consider a lot of things like the clinical part, the radiographic, microbiological, immunological aspects when arriving at the diagnosis and the treatment of the periodontal disease. In the current technology-driven era, there are a lot of advancements in dentistry and in periodontal diagnosis. These advancements not only help us in diagnosing and treating, but also in predicting the clinical outcome. Now, the goals of the periodontal diagnostic procedure, it provides as a very useful information to the clinician on the periodontal disease type, the location, and the severity. Now, this information forms the basis for us to plan and monitor the disease during the recall visits. According to Armitage, the purpose of the periodontal diagnostic procedures can potentially serve the five purposes, that is monitoring the patient, screening, diagnosis of the specific periodontal diseases, identification of sites or subjects at increased risk, and the treatment planning part. Now, all these definitions and all we are included because of the exam oriented topics, and these should be a part of your answers. The diagnosis is defined according to the glossary of periodontal terms as the correct determination, discriminative estimation, and logical appraisal of conditions found during examination as evidenced by distinctive marks, signs, and characteristics of the disease. Whereas a periodontal diagnosis is defined as recognizing a departure from health in the periodontium and distinguishing one disease categorization, or etiology from another, based on the information obtained from the medical and dental histories, clinical and radiographic examination of the patient, and the laboratory findings. Now, with the clinical aspect, we have the provisional diagnosis, the therapeutic, or the differential diagnosis. Once we send the patient for the diagnostic part, we can arrive at the final diagnosis. The diagnostics can be referred to as the tools or the procedures or technologies that are used in the determination of diagnosis, which is used to predisposing risk factors, identify early disease, and specify the type of the disease. The efficacy of the diagnostics can be determined as the gold standard. It should be accurate, should be sensitive and specific, and should have a positive and negative predictive values. The most important use of the diagnostic test is to detect the disease presence, evaluate the severity, and assess the actual response of its completion, evaluate the response to the treatment, and predict the clinical course and the prognosis. According to Chappell, these are the ideal criteria for the diagnostics. And Page has referred the diagnosing periodontal disease into several decision nodes. It consists of three levels. The first level, wherein we are able to diagnose the health versus the disease. The second level, we are able to differentiate between gingivitis and periodontitis. And the third level, we are able to classify the different types. And lastly, the decision to determine whether the disease is active or arrested or in remission. Now, this becomes a very important part when you're treating the patient, because the periodontal disease is site specific. It can be active or arrested in certain sites. So depending on all these factors, if you have a diagnostic tool which can identify these factors, then treating the active or inactive lesion can be done in an appropriate way by the clinician. Now, to get into these things, there are a lot of diagnostic aids which we are already using that we can categorize as the traditional ones. Now, the technology driven or the advanced diagnostic aids play a very, very important and major role in the present day, where we are managing the clinical part as well as the other aspects of the disease. Traditionally, we were the treatment aspect, the diagnosis and the treatment aspect were dependent morely on the clinical attachment loss. You send the patient for an X-ray check, what is the bone loss, and it usually provided an historical evidence of the damage, which is already existing. We never knew how it started and what are the causes. We had to evaluate that. Now, yes, we have been working with those things, and they were quite easy to use, cost effectively, and proved clinically good information. But they were not entirely suitable for monitoring the progression of the periodontal disease. Now, these drawbacks of the traditional diagnostic procedures are intended or expected us to use the more advanced diagnostic aids. Now, this chart is the most important of all the slides today. this has to go as a printout onto your wall. Now, this summarizes all the diagnostic aids, and this will form the basis for your essay question. The diagnostic aids can be classified as the clinical diagnostic aids, the radiographic aids, the microbiological aids, genetic analysis, and the biochemical, which is the characterization of the host response using the saliva or the GCF. Now, most of the questions in this topic revolves around this chart. Now, there are questions, the short notes which they ask about the probes. There are short notes which they're asking about the cross-sectional 3D imaging, or they ask about the BANA or the ELISA test. Now, once you have a picture of all this, the essay question will be quite elaborate and up to the point. Now, talking about the traditional or the conventional diagnostic aids, the periodontal probing forms one of the most important aspect of the treatment or the diagnosis. The probes were classified by Filstrom and Watts into different generations. Now, each generation overcomes the disadvantage of the previous generation, and this is the National Institute for Dental and Craniofacial Research, which has defined 8 criteria for overcoming the limitations of the conventional periodontal probing, where they compared the conventional with the NIDCR criteria. Now, this chart gives the advantages and disadvantages of all the generation of probes. Though the first generation, the manual probing, is considered as the gold standard method, there are certain disadvantages. To overcome that, we have the second, third, and the fourth generation. But in all that, again, there is an inability of getting the 3D information. We cannot visualize what is the bone loss. We cannot actually see what is there underneath. To overcome the disadvantages, they also use certain imaging technologies for periodontal pocket evaluation. These included the use of CBCT based imaging with a radio opaque contrast agents, or the optical coherent tomography, photoacoustic imaging tomography, endoscopic capillaroscopy, and the MRI. But again, all these techniques just gives again, all these techniques, there are some techniques wherein there is the patient is exposed to more of ionizing radiations, and they are quite expensive, and pocket evaluation is done at a very regular interval. So how many times are these imaging techniques to be are the subjects the patient to be subjected to? Now, to overcome these things, we have the 3D technologies, which I'll be talking about. Now, these are some of the, I found these articles to be quite interesting, and comprises or gives a gist of the manual and the digital probing or the latest advantages. To conclude about the probing, despite all the limitations, the manual probing is still the standard of care for periodontal diagnosis globally. However, with the increasing technology updates, now this method also has to get digital, and it has to be used in a technology driven way. Gingival temperature, yes, it is another parameter. Now, Hafji and Kung has done a lot of studies. The periotemp probe is the instrument which is used, and it detects pocket temperature difference of 0.1 degree centigrade from the reference subgingival temperatures. Now, in his early studies, the Hafji has conducted, he has concluded that whenever there is an increase in temperature, there is a future loss of attachment. He is also told that the increase in the number of microorganisms are found at these sites. But the possible explanation is that either the microorganisms increase the temperature by initiating the inflammatory process, or the increased temperature provides environment favorable for their growth. However, the influence of the pocket depth on the temperature is still not clear, and further studies are needed to demonstrate the accuracy of this device and its utility in the clinical diagnosis. To conclude, subgingival temperature has a good specificity but poor sensitivity when considered alone as a marker for the progressive periodontitis. Tooth mobility, conventionally, we use the metallic instruments, the blunt end, and we check it. The advanced way is using the Perio test, which gives certain ranges wherein we can confirm whether the tooth is firm or it is mobile. The Perio test also can be used to check the mobility of the implants. There are other methods like the laser vibrometer, the resonance frequency analysis, and the dental mobility checker, which can be used for evaluating mobility. Halometer is one of the chairside devices to assess halitosis. The gold standard is still the gas chromatography. The other methods are BANA, which is one of the favorite. There are other portable monitors like the Tanita breath analyzers, the electronic nose, and the HaliCheck, which can be used to check halitosis. Occlusion plays a very important role, as Dr. Vijay was discussing. Yes, it definitely determines the progression of the periodontal disease. Now evaluating occlusion and the occlusal discrepancies is again a major concern, and it needs technically sound knowledge and the experience of the operator to analyze this. Now, to overcome all those things, now we have a T-scan, which gives a digital printout about the occlusal discrepancy, which can be attended and corrected. It is also useful in educating the patient as a tool to enhance the comfort and increase the case acceptance. To determine the calculus, we use the detector, um, which 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 can be used as an handpiece, but yes, the tip is quite bulkier for the penetration. That is one of the disadvantages, and yes, it comes with a heavy price tag. Dental endoscope or the perioscope is quite an useful tool in evaluating or visualizing the deeper parts of the pockets, which are usually not visible. That gives an idea about the internal structures. Coming to the radiographic diagnosis, the conventional way of evaluating is using the periapical radiographs, the panoramic view, the occlusal, and the cephalometric radiography. But because of the poor sensitivity and variation in the projection geometry and improper visualization of the structures, maybe we can't do that as a 3D imaging. We can switch on to the advanced methods, but there are certain advanced 2D imaging techniques in the term of microradiography, zero radiography, stereoscopy, and scanography, nuclear medicine, iodine 125 absorptometry. But again, these are 2D imaging techniques and comes with a good price tag, whereas you can stick on to the older conventional methods instead of these things. Even the subtraction radiography and the digital subtraction radiography comes under that category itself. Now, these things can be overcome with a cross-sectional 3D imaging. As an examination point of view, the CBCT is quite important, because that has been also asked as a short note in one of the RGHS papers. Now, when you are dealing with CBCT or when you're writing an answer on CBCT, always write the principle of the CBCT, what are the advantages and what are the different sections, and how it can be used in the periodontal diagnosis, as well as in the implant planning part. One of the major advantages of the CBCT is the extensive hard tissue knowledge what we get, but yes, the soft tissue part is very ill defined. If you're looking for a soft tissue uh, evaluation, then the MRI should be a better option. We have certain non-invasive techniques in the form of ultrasound. But one of the major uses that I found is with the ultrasound is to assess the gingival thickness and the biotype. And the major advantage of this is it's a non-ionizing radiation, and these are just sound waves, and it is not very expensive for the patient also. But yes, there is a disadvantage that it has a limited use because of the anatomical uh, complexities of the head and neck region. It can be used only in the superficial areas, and you have to get a little trained in doing that and reading the images. These are some of the this is one of the papers where they have used the conventional imaging. They have compared it with the advanced CBCT imaging. Yes, the conclusion is that the CBCT is better than the conventional ones. Whatever it is, we cannot replace the conventional 2D imaging, at least as a chairside protocol. But yes, whenever in doubt, we have to go in for the additional 3D imaging techniques as the CBCT or the MRI to plan and diagnose the cases. Going on to the next top aspect is the microbiological. Important methodological issue for the application of the microbiological testing is which site should be sampled, and are these sites representative of the entire dentition? Since the periodontitis is site specific, now if you take a sample from one site, will that represent the whole thing? That is always a is of question. But yes, it can be used to determine the periodontal sites at a higher risk for the active destruction. It can be used to identify putative pathogens, to monitor the periodontal therapy, and also in aid in the treatment planning. There are different methods of the microbiological examination. One is the culture, the microscopic methods, the immunodiagnostic, the enzymatic method, and the diagnostic assays based upon the molecular biology techniques. The bacterial culturing is referred to as the gold standard method even today of all the microbiological diagnostic aids. But it has a lot of disadvantages, like it requires strict sampling and transport conditions, and it can grow live bacteria only. Some pathogens are fastidious and difficult to grow. It has a very low sensitivity and quite time consuming and expensive. An alternative for the culture method is the dark field or the phase contrast microscopy. But again, this has a disadvantage of unable to identify the non-motile pathogens and unable to differentiate among the various species of Treponema. To overcome this, we have the immunodiagnostic methods, which is quite popular among most of the clinicians, especially the ELISA. The various procedures also include the direct and indirect immunofluorescence assay, the flow cytometry, membrane assays, and the agglutination test. Now, where do you indicate the immunodiagnostic methods? Now, if the organism is unable to cultivate on the artificial media, if the organisms fail to survive the transport to the lab to detect the individual bacterial species or to detect, preserve, and relative proportions of selected bacterial species. Now, these are the indications for the uh, immunodiagnostic aids. Now, coming to the first of that, the immunofluorescent assay, it is used to detect the bacterial species that are genetically distinct. They help in identifying the target bacteria. There are basically two types. One is the direct and the indirect methods. One of the earliest studies by Zambon et al. Indicates that higher sensitivity, this method has showed higher sensitivity when compared to the culture method. So when there is a limitation for the culture method, you can go in for an IFA. Flow cytometry, yes, it's a good one to for the rapid identification of the oral bacteria, but because of the cost factor, it is not very widely used. Now, this is the most important and the very popular means of the assay, that is the ELISA. These methods have shown a very high sensitivity and specificity than bacterial culturing for the detection of the target microorganisms. Now, ELISA is also asked as a short note. So when they are asking that you need to elaborate uh, on the on the steps of the ELISA, okay, the basic types or the non-competitive and the competitive and the inhibition ELISA, wherein your non-competitive have the direct, indirect, and the sandwich technique. Now, as a short note, you need to elaborate a little more in detail for this particular part. Now, there are certain limitations of the ELISA. The results may not be absolute, and the antibody may not be available. There are certain false positive and negative results also, but still we have to count on the advantages where it's very easy to perform, quick, widely available, and has a long um shelf life. There are a lot of studies. There are loads of studies in the periodontal literature wherein they have used the ELISA to assess the various microorganisms and the various parameters. Coming to the agglutination test, you have the slide agglutination and the latex agglutination. It is used to detect the microbial and the viral infections, autoimmune diseases, hormones, drugs, or serum proteins by agglutination reaction of the antigen antibody. The enzymatic methods it approach for the detection of selected bacterial species to look for the presence of an enzyme that is unique to one or more of the clinically relevant species. One of the most common and the favorite one is the BANA test. It hydrolyzed by the trypsin-like enzyme produced primarily by Treponema denticola, bacteroids, forsythis, and the Prophenomonas gingivalis. One of the earliest studies, Sokransky and Hafaji, involved around 1,000 plaque samples taken from over 100 patients. They found the BANA positive species. They are the T. denticola, the Prophenomonas gingivalis, and Bacteroides forsythis to have the highest prevalence and to be present in the highest level compared to over 40 other plaque species that were evaluated by the DNA probes. Now, the diagnostic kit, that is the Perio scan, has been developed using the BANA hydrolysis reaction to detect the bacterial trypsin-like proteases in the dental plaque. Various studies have been done where the BANA test is used as a risk indicator for the periodontal attachment loss. But as it is having a lot of advantages, like it is rapid and inexpensive, okay, and it can be used as a chair-side evaluation method. But as the disadvantages being the false positive results, and it can detect only 3 pathogens and does not detect sites undergoing active destruction. This comes as a major disadvantages of using this technique. But yes, again, you have a lot of articles uh, which encourage the use of BANA as a chairside test for the proper diagnosis of the periodontal conditions. Now, the diagnostic assays based on the molecular biology can be categorized as the nucleic acid probes, the restriction endonuclease analysis, DNA checkerboard, and the PCR. Now, there are various studies which indicate the positive use of all these methods, whether it is a nucleic acid probes, the DNA checker, or the PCR. When they are used, the DNA probes, they're quite useful for the detection of the pathogens, the REA for certain kind of Echinella corrodens. There are the fish or the fluorescent in situ hybridization method can also be included under this category. When you compare all these things, the checkerboard DNA, hybridization technique, uh, now these things which helps the comparison of the microorganisms as well as the hello. Hello, am I audible?
Yes, you are audible, doctor.
Okay. Now, this enlists the various methods for identification of the pathogens, the microbiological and the chemical investigations, the culture methods to the oligonucleotides or the DNA hybridization, and what exactly does the description. Now, this has to be noted as a part of your examination protocol. Now, going to the advances in characterizing the host response. The assessment of the host response refers to the study of mediators by immunologic or biological methods that are recognized as part of the individual's response to the periodontal infections. Now, these mediators are called as the biomarkers. The definition of biomarkers is important. A biomarker or a biologic marker is a substance that is objectively measured and evaluated as an indicator of the normal biologic process, pathogenic process, or the pharmacological responses to a therapeutic intervention. Curtis classified the biomarkers as the indicators of current disease, predictors of the future disease progression, and the predictors of future disease at currently healthy sites. Now, the candidates for biomarkers in periodontal disease include the bacteria and their products, the inflammatory and the immune products, the enzymes released from dead cells, the connective tissue degradation products, and the products of bone resorption. These are some of the commercially available kits to assess the microbiological markers, the Evalucite, the Omigen, and the Perioscan. Perioscan is quite popular, as I told you, which is used as a chairside kit utilizing the BANA test. The advantage of these will be, they are it's results of the chairside kits are available in very short duration. Some appear to be predictive of the disease activity in longitudinal studies. Now, the chairside kits provide the visual result, which can be shown to a patient as a method of motivation, and the acceptance of treatment is also quite good when we do that. Now, coming to the inflammatory and the immune markers, the inflammatory and the immune host response, along with the direct effects of the bacteria, cause most of the tissue destruction. Now, these pass into the gingival, clavicular fluid, and saliva, and are thus available for analysis. Now, I am just enlisting the initial or the important studies with the results so that you can go back and elaborate on these things or include it in your answers. The hydrolytic enzymes of tissue origin. The inflammatory cells contain the destructive tissue enzymes within their lysozymes, which are normally used to degrade the phagocytosed material. Again, these enzymes are capable of degrading the gingival tissue component also, and this can be enlisted as the proteolytic enzymes or the hydrolytic enzymes. Some of the host-derived enzymes following the cell death will be the aspartate aminotransferase, the lactate dehydrogenase, alkaline phosphatase, the B-glucuronidase, and the matrix metalloproteinases. These are some of the studies which were based on the studies on the enzyme-based biomarkers. The Chambers and the Lancet study are quite important among this. The markers of the connective tissue origin, the breakdown products which were enlisted, and the components from which the breakdown products have been obtained. The markers of bone resorptions include the osteonectin, the bone phosphoprotein, osteocalcin, collagen, prostaglandins. The advantage of evaluating these bone resorption markers is that identifying the presence of disease, but you cannot actually predict the risk part, and also the chairside diagnostic tool is not available. Now, this chart becomes a very, very important chart for your Viva. Okay, this enlists what all the microbiological, the biochemical, and the genetic test and their trade names, and what do they evaluate. Now, these can be listed, and these individual things can be asked as Viva as to what it is. Now, there are other kits for the biomarkers which are new or which are a little uh, um, new in the market, that is the noggin and the infusion. There are certain advances in the genetic assessment, like as the Corman and colleagues have suggested, the genetic susceptibility test for the interleukins. Now, these are some of the articles which you can uh, refer to have a better idea of these things. Now, there is an important or the latest concept of point of care diagnostics. Now, any rapid chairside test which is used to diagnose the periodontal disease, it can be categorized as the point of care diagnostics. Now, these include the lab on a chip system, which I'll talk in the next slide. The oral fluid nanosensor test or the OFNA set, which detects oral cancer. Visilite kit, it's again for oral cancer. Electronic taste chips to evaluate the CRP levels. The Aura Quick, which is the FDA approved one, which we take the oral swab and send for the detection of the HIV. The integrated microfluidic platform for oral diagnostic, which evaluates the salivary biomarkers. These are um, quite new, and this concept we term it as the point of care diagnostic. The lab on chip, now you get a you evaluate the GCF or the saliva samples, and you can check for the multiple biomarkers. That is one major advantage of the lab on chip, where with a single sample of the GCF or saliva, you evaluate the multiple biomarkers in that. We have the dipstick glucometer, which you can use it to evaluate the sugar levels using uh, the clavicular blood. We also have the diagnostic rinse for periodontal disease, which gives the extent of the periodontitis, either it is a moderate or a advanced one. And now to conclude, the focus on the disease prevention is mainly on the disease prevention, early diagnosis, and intervention to minimize the treatment, which enables the desirable outcomes. The diagnostic modalities available today expand greatly on the foundation of a comprehensive visual assessment, which has been and will be the cornerstone of the diagnostic process. In the future, combination of tests rather than one test for one single aspect of periodontal disease. Now, this again gets to the fact that periodontitis is a multifactorial disease, and we have to consider all the possible aspects of the disease when we are planning for a treatment and arriving at a diagnosis. Now, for that, all the advanced diagnostic aids will be of quite useful. To conclude, very few diagnostic methods are promising in the clinical practice. Even today, there are not many chairside. As a clinician, we don't use many of the chairside tests as a routine practice. Okay, we need to work accordingly on the treatment of the periodontal disease. And the more we know about the pathogenesis of the periodontal disease, the more we realize that we still don't know a lot. So the pursuit of the best diagnostic aid for the periodontal disease will go on, and this becomes a area of research for the young graduates. Now, these are the questions from the RGHs which have been asked in the uh, previous examinations, the 100 markers, the 20 and the 10 markers. Now, again, to summarize, now, if it is a 100 marker, if it's the essay question, then yes, you need to, this will be your outline. And again, this chart is the most important aspect of your study in this topic. Now, this encompasses all the different methods, and individually for each questions, as I mentioned before, for short notes, this you can also use as an outline. I am wishing all the students a good luck for your future. Thanking you all. I also take this opportunity to thank the organizing committee of BAP. I thank my mentors, Dr. Faizuddin sir and Dr. Ramesh sir. I thank my principal, Dr. Hemlata ma'am, for providing the platform of our college in conducting this program and always being a backbone and a support for all such events. I also take this opportunity to thank my teachers, Dr. Shanti Priya and Dr. Sarita, for giving me an opportunity to uh, be on this platform. My colleagues and staff members who have been always helpful in as of now in conducting these programs, and last but not the least, all my PGs who have been working beyond their comfort zone to uh, get this program into a very nice stage. I thank you all, and I wish you all bright future. Thank you.
Thank you, Dr. Lalit, for very effectively condensing such a vast topic in such a limited period of time. Yeah. Before we wind up, I request you to answer few of the queries that we have received. One
Yeah.
is that can you use advanced imaging technique as a substitute for conventional probing?
Both the speaker and the moderator, come on to the video mode, please.
My video is on.
Yeah.
There is some issue with the video here. My audio is clear?
Yes, it's clear.
Yes.
Okay. So yes, we cannot substitute the conventional probing techniques. Yes, we can always add on the advantage of the advanced diagnosis, like using the third generation probing. But again, the for a clinician, it adds on to the economy part of it. But as a yes, but we cannot replace the manual probing. It still has the gold standard view.
Okay, so we can use this as an adjunct to our
Yes,
manual probing, right?
exactly.
And another thing which I would like you to answer is how feasible are these point of care devices in a clinical setting?
Now, these are the next things that we have to be equipped in our practice or as clinicians. The point of care diagnostics, like using a small chairside diagnostic tool. Now, the patient, if you take his point of view, from a long time, they are just seeing the mouth mirror probes and the other, small diagnostic aids what we do. Now, these will form as an adjunct in educating the patient, plus also in predicting the diagnosis and the future uh, prognosis for the patient and the disease process.
Yes. So it saves a lot of time for the patient as well as the clinician also, right? If we have
Exactly,
point
and
of
it
care
educates the patient also in a much better way.
device. Thank you once again, Dr. Lalit, for that wonderful lecture. I request you to accept a token of appreciation from the organizing team.
Thank you, Dr. Nisha, and thank you all the organizing committee.
Thank you.
Thank you, Dr. Nisha, for accepting the invitation for moderating this session for this program. We thank you for your contribution. Please accept this appreciation.
Thank you so much. I once again thank the organizing team for considering me as a moderator. Very happy to be a part of this wonderful program. Thank you. Thank you, Dr. Fausia.
Thank you, Dr. Nisha. We have been much ahead of time. Now the break. We shall begin the following session on the scheduled time, that is 1:30 p.m. Please log in at 1:30 p.m. Thank you.